Good afternoon. Wow. Welcome to the Kavli Foundation Special Symposium. It's a quantum world, and indeed it is, here at the Global Physics Summit. I'd like to give special thanks to the Kavli Foundation for making it all possible. Well, the scientists made it possible too. This year marks a historic convergence as we combine the March and April meetings into a single groundbreaking event here in Anaheim. We are bringing together close to 15,000 members of our global physics community, plus many more in 17 satellite sites around the world. Going off script here a little bit, I had some trepidation about this meeting for various reasons, but my unscientific, somewhat statistically significant assessment here on almost the last day is that it's gone pretty well. And I think part of it has to do with the venue, but also part of it has to do with the amazing enthusiasm and flexibility of our membership. Quantum science and technology are at the heart of this meeting. The field has undergone a remarkable evolution over the past century, transforming from pure physics to practical applications and revolutionizing our world. From advanced sensing to secure communications to quantum computing, quantum technologies are reshaping our understanding of the possible. And today's symposium is a testament to the great spirit that drives the field. Before we hear from the speakers, I'd like to take a moment to highlight something special that's happening on the global stage. Last month at its headquarters in Paris, UNESCO launched the International Year of Quantum Science and Technology. This initiative, led by APS and designated by the United Nations, is bringing together scientists, educators, and policymakers from around the world to celebrate the profound impact of quantum science on research, technology, and society. I'd like to share with you the opening video, first shown at UNESCO in Paris. Let's take a look. So as you can tell, inspiring and educating the younger generation is a key to the IYQ goals. I hope that you too are inspired by the incredible potential of quantum science, and I encourage you to join in the global celebration. I also want to recognize that many of you may be concerned about events in Washington. Whether you live in the US or abroad, the administrative actions are targeting the global scientific enterprise. We need to help lawmakers understand the transformative impact of federally funded research, how it creates local jobs, supports local economies, and fuels innovation, which is fundamentally important to the long-term health of the US economy. APS has launched a nationwide advocacy campaign calling on Congress to protect funding for basic research. I encourage you to participate. Details are, of course, on the APS website, very easy to find. I would now like to introduce the chair of today's session, the Global Physics Summit Program Co-Chair, Professor Ken Brown. Dr. Brown is an APS Fellow and a Distinguished Professor of Electrical and Computer Engineering at Duke University, where he also serves as the Associate Director of the Duke Quantum Center. His interdisciplinary work has significantly advanced our understanding of quantum information and computation. His leadership and dedication to the field make him an ideal chair for this symposium. Please join me in welcoming Ken to the stage.
All right, good evening. Welcome to this Kavli session. I'm super excited about it, where we have a great lineup of speakers which connect all of quantum aspects across what I think of many March meeting disciplines, from questions in biology all the way to where our ideas of quantum computers can even interact with things like quantum chromodynamics. We are also have joined by people online. So for the online people, the reason I'm sitting down in front of this computer is to see your questions and answer your comments. So with that, I would like to introduce our first speaker, Jim Werner. Jim Werner is a scientist at Los Alamos National Laboratory. He's famous for applying quantum optical approaches to many interesting questions in biology, and it's a great pleasure to have him here. Jim.
Okay, well, thank you, Ken, and thanks for the organizers, and thanks for everyone in the audience coming to hear this talk. I'm going to tell you about near-infrared quantum ghost imaging in living and unperturbed plants. And so before I get into the quantum aspects of it, I would just want to talk about plants for a bit. And plants can actually do something pretty amazing. You know, they can take sunlight, water, air, and dirt. And with that, starting with that, all I could make is mud. But plants, you know, they give us food that we can eat. They give us materials and fabrics that we can wear and use. They also take single carbon in carbon dioxide and make higher carbon element macromolecules out of it, things that we can potentially burn as fuel in cars. And so plants can do this pretty amazing thing, which is photosynthesis. But, you know, so I just want you to know that plants are important and that they do these pretty amazing things. And they're a complex system. Photosynthesis has proteins, it has specialized cells, it has many things working together in concert. And I think one of the best ways to understand complex biological systems is microscopy and imaging. And so, you know, I think microscopy and imaging are very powerful and useful techniques in a wide range of sciences. Because I'm giving this talk at APS, I actually chose an example from astronomy rather than biology, because I think this might resonate more with the people in the crowd. And also, it was the best example I could think of a picture that really made national press and even lay people got to see. And it's the picture of a black hole backlit by a hot lit plasma of M87. Later, they did the Milky Way, the black hole in the center of the Milky Way galaxy, that team. You know, but I really think it drove home a picture is worth 1,000 words. You know, news articles were showing this and news reports were showing this picture. They weren't reading the first 1,000 words of that paper. And so there's also, you know, in terms of, you know, microscopy being important, I'll just say there are many fairly recent Nobel Prizes related to microscopy development of GFP, development of super resolution microscopy, quantum dots. Part of that was for biological imaging applications. So, you know, I want you to know that plants, I think, are important, and that I think microscopy is an important way to look at complex systems like plants. But when you want to use microscopy applied to plants, you run into certain challenges. One is, you know, key one is plants respond to light. We all know about sunflowers, you know, heliotropism, following the light. But there are other light-dependent and light-independent processes in photosynthesis itself. There are other macro changes to plants during night. You might know there are holes in the leaves of plants called stomata that are used for gas exchange, bringing CO2 in and releasing oxygen. Those are open during the day when you're doing photosynthesis for gas exchange, but at night they close up. so you don't release water from the plant. So there are a lot of things. Plants respond to light, and anytime you want to do laser microscopy or any type of imaging with light on them, you're potentially changing the system from its basal or resting state. So plants respond to light. The other problem with fluorescence microscopy, as I typically do it, you typically, use a fluorescent label, and any type of label, whether it's a GFP or a dye, label antibody or whatever you're using, you have the potential of that label to perturb the biomolecular dynamics that you're studying. So, pictures or microscopy is useful, incredibly useful for complex systems, but there's some challenges applying microscopy to plants. And I'm going to tell you how with quantum-enabled imaging, we can kill two birds with one stone. And so the two birds we can kill are the need for labels. We can obviate the need for any labels because we can image in the infrared where vibrational absorption gives us contrast. The other thing we obviate is the need for high laser fluence. We can actually do this with very, very low light levels. We're using a light flux, which is about 50 times less than a moon-filled night. And so I would say, this is just a thought experiment about using quantum methods to kill birds. I'm doing all I can to keep the spirit of Erwin Schrödinger alive, who famously thought about ways you could use quantum methods to kill cats. So, moving on, what we're doing is a technique called quantum ghost imaging. And I think this is a really cool technique and useful, but I'll tell you how this works. You take a pump laser at high energy, and by high energy, I mean it's bluer than the other beams in here. You can take that one photon at high energy and split it into two photons at lower energy. These are typically called your signal and idler photons. And you do that in a nonlinear optical crystal by a process called spontaneous parametric down conversion. In quantum ghost imaging, the signal beam goes through free space. He's relay imaged onto some type of imaging sensor or detector. There's basically nothing in this beam other than optics to relay the crystal onto your imaging sensor. Your other photon of that pair, the idler, here in this example, goes through an object you want to image, and then whether that photon made it through that object, that you detect with a single element detector that's called a bucket detector. So there's no imaging on this bucket detector, just did we see a photon or not? And then what's neat about ghost imaging is if you correlate photon arrivals on this imaging sensor with photon arrivals on this single element bucket detector, you can computationally recreate an image of this object, even though the photons hitting this sensor never went through your object of interest. And I don't know if you're anything like me in the audience, but the first time I saw this, I was like, how in the heck could this possibly work? And I think the easiest way to understand this is in terms of conservation of photon momentum in this down conversion process. That, your pump laser is well-defined momentum going left to right. And if your signal photon goes up a little bit, like it's exaggerated here, your idler photon has to go down a little bit because h bar k of your signal has plus idler has to equal h bar k of your pump. So, when this guy goes up a little bit, you've measured his k vector, where he hit on this detector. And so the k vector of the photon which went through here. And through this correlation, you can combine sort of where the spatial location is here with the transmission information of this photon. And it's really the combined information from both of these paths that you're using for image formation and ghost imaging. So I think ghost imaging is a really cool way to image something. You can image objects with light which never went through your sample, but I also think it's potentially a very useful way to image things. I guess I forgot that I do think an interesting analogy is quantum ghost imaging. It's sort of like a game of Battleship with entangled photon pairs. So your imaging detector might see a photon at coordinate G4, and then you ask your bucket detector, did you see that? And if he saw that, it's a hit. He saw that photon, in this coordinate space, the k vector which corresponds to this location, that photon made it through, so maybe your sample's transparent at that coordinate spot. Then you can try a different coordinate spot. Did he not see it? Then it might have absorbed it. And you can really build up a map of the transmission in this other arm. So again, I do think it's a really cool way to image something. You can image objects with light which never interacted with your sample. But I also think it's potentially a very useful and powerful way to image systems. And in particular, I personally see two key strengths of quantum ghost imaging. One is that your photons do not have to be at the same color or energy. So it can be probing at one wavelength, say the near infrared, where things like vibrational absorption give us contrast. It can be doing your image detection and image formation with visible photons. Visible photons are higher energy. We have really good imaging sensors in the visible range. So I think that's a key strength, is we can do wavelength shifting to start to do imaging at near infrared wavelengths with visible detector technology. The other key strength I alluded to is that because when you create entangled photon pairs by SPDC, these two photons are created, nearly simultaneously. And so because of that, you can, if you're doing coincidence detection with time-resolved detectors, you can measure and have a very narrow time window for correlation of these photon pairs, and you can beat down a lot of other sources of noise in your measurement, including stray light, including detector dark counts, because those are uncorrelated between your two detectors. So you can do this type of imaging at very, very low light flux. So these are the two key strengths of quantum ghost imaging. And I think they're very useful for plants in particular because they're light sensitive. So when you talk about what's in a plant and what absorbs in the near infrared, this is a chart we compiled from a number of references. But, I turned off the deadly blackout button instead of the laser pointer. So I want, there's water, proteins, lignocellulose. These are things that make up plants. I want to turn your attention to the curve for water. So there's a water absorption peak, or a little bit less than 1.5 microns. This is a vibrational overtone of water. And we figured, let's start here and do ghost imaging here. A few reasons for this. I'm a physicist. I figured I don't know much about plants, but I know water is fairly important for their growth. But also 1.5 microns, as people may know, is close to a telecom wavelength. So lots of people have been doing things like quantum key distribution there. And so I figured, let's start here. I think it's going to be easy. The other thing I like about water is it's far from a trace element. It's present at like 56 molar in most things. And if you can't detect that, you got problems. So we started there, and our funding comes from the Department of Energy, as I'll tell you at the end of this talk. But we propose to do this in 2 biofuel plants, or crops which can potentially be turned into biofuels, sorghum and camelina. We chose them not only because they're 2 viable candidates for biofuels, they use different ways of photosynthesis and carbon storage. Sorghum is what's known as a C4 grass. Camelina is what's known as a C3 plant. These numbers have to do with the number of carbon atoms in the first stable intermediate in the Calvin cycle. And so we thought we could do potentially compare contrast of how these plants respond to different environmental stress and do photosynthesis with these quantum-enabled imaging methods. So this is a more realistic diagram of how these experiments go. We bring in a pump laser. We create entangled photon pairs with this SPDC process. We recollimate the beam with a parabolic mirror. Then we send our visible light through free space onto an imaging detector, NCAM. I'll tell you a little bit about this because it's a fairly special imaging sensor we're using. Then the infrared beam goes through our sample of interest, and then we detect him on a single element indium gallium arsenide single photon counting avalanche photodiode, or SPAD. And then we do our correlation post-process. This is what our pump laser frequency spectrum looks like. Wavelength spectrum of our signal beam, which is about 540 nanometers. We're doing our imaging. And then we're probing at this vibrational overtone of water out here at about 1.5 microns. So I did want to spend a little bit of time telling you about this detector, NCAM. It's currently called NCAM. This detector belongs to a class of sort of photocathode-based imagers. These used to be at Los Alamos called remote ultra low light imagers, RULI sensors. And I would say the thing about these sensors is it's unlike, we're calling them cameras, but it's unlike any camera it has no pixels, it has no frames. It detects single photons over about a 1-inch round imaging sensor surface and can tell you where that photon hit with 100 microns accuracy in X and Y. It can tell you when it hit with about 100 picoseconds accuracy. So the data out of these things is just X, Y, and T. How they work, I do have time in this talk. That, how these things work is you take a photon, hits a photocathode, generates a photoelectron. That photoelectron gets amplified 10 to the 7 times through an MCP stack. That MCP stack then hits an anode array. There are 2 wires in NCAM or RULI, which one wire serpentines through and giving you sensitivity in X. The other one serpentines through, giving you sensitivity in Y. And the time delay or difference between when that electron cloud hits on either end of that anode structure tells you, gives you your Y position for the 2 Y lines and your X position for the 2 X lines. And so I would say this class of detectors, NCAM or RULI, they were developed at Los Alamos National Laboratories for a lot of national security applications, things like LiDAR. When we started this project, having that technology at Los Alamos really, I think, allowed us to separate ourselves from a lot of other people trying to do ghost imaging with this camera technology. I would say when we first wrote this grant, you could not buy one of these things, but now you can buy photocathode-based imaging sensors from a German company, Photon Score and LinCam, and we do have some of those. The great things for this quantum ghost imaging, for this NCAM or LinCam or photocathode-based imagers is, again, this exceptional time resolution. That this is a 1-inch round image sensing surface, and you can tell photon arrivals at 100 picoseconds. So I've lived my life doing like time-correlated single photon counting with SPADs, and you're always getting the smallest detector you can for the best IRF. And here, this is just a histogram between our bucket detector and our camera, and we have about a 200 picosecond full half max, which is the combined RRF of both. That just blew my mind that you could get this with an imaging sensor that's 1 inch across. So because, again, how well you can define when these photons arrive, that is really how well your signal to noise goes, is 1 over that window width, how well you can precisely time this photon pair. So the other great thing about these detectors is they're very low dark counts. And so, this correlation can beat down noise, but it's great if you have a very low noise detector to begin with. You're talking about 100 counts per second at room temperature per millimeter squared, about 4 counts per pixel sensing element spatially. So we built this quantum ghost imaging system, and we started imaging sort of simple targets. The first thing everyone asks is the first thing you imaged, and I'm like, a razor blade, because you basically block half your beam, and can you see that? It's a very simple target to image, but also razor blades and knife edges are useful in optics because how sharp that knife edge is tells you something about whether you're in the focal plane and what your spatial resolution is. And so our spatial resolution is on the order of 15 microns. We're using relatively low NA optics for all of this. We're also in the infrared. And that's about 15 micron spatial resolution over a field of view, roughly 300 by 300 microns. So after you do like a razor blade, you start making sort of cutesy test binary targets. And by binary targets, I mean you're basically blocking the beam in part of your path and letting 100% light transmission through and the rest of it. So, we were doing quantum ghost imaging, so we decided we'd do a ghost from Pac-Man. And I was glad to see Pac-Man is alive and well in APS in the exhibit hall. If you've been there, there's this 80s arcade. There's actually have a few different versions of Pac-Man there. But also, if you're doing any type of imaging thing, your institution is going to want you to image their logo. So we also did the Los Alamos National Lab logo, which you've actually seen down here for most of my talk. But, if you want to move beyond sort of simple binary test targets, basically in the past, when people, I had a conversation with one of my former postdocs this morning, and we were talking about how in every imaging paper, everyone does like all these test things, and then they throw in one biology example at the end. And this is sort of the biology example at the end for 2 sort of papers I could find on actually using quantum ghost imaging to image biological materials. One is from Boyd and Paget's lab a number of years ago, and they imaged a wasp wing. And I don't know if you've ever seen a wasp wing or a fly wing or a dragonfly wing, but that thing is pretty much flippin' transparent. It's like 1 cell layer thick. Doing transmission imaging through that is not that difficult. Another group in Germany recently actually was working at a very similar wavelength to us, this 1.5 micron line, spectral area, and they imaged a single layer, epidermal layer of an onion skin. And so that's sort of like the plant equivalent of a wasp wing. That's like one cell layer thick. It's pretty much flippant transparent. You can see through it. It's not a very challenging sample to look through. So the problem we faced was when you throw in something like a blade of sorghum, a leaf from a plant, you go from 10,000 correlated counts per second, for integration time, down to 100. So you're basically putting an ND2 filter in your system. So you're now trying to image with an ND2 filter in front of your camera, more or less. But because we have such excellent temporal resolution with this camera, we can beat down and integrate for longer because we can beat out all of this sort of grass and cabbage and less correlated photons to the side of our correlation peak window. And so that really enabled us to image live plants in transmission mode. So this is a sorghum plant in our quantum ghost imaging microscope. It was just sort of had its leaf growing that way, and we just taped it to a slide here, and we're imaging not through the glass or through the tape, but really just to the side of it. So we're imaging sort of this leaf through free space. And what we found imaging at this 1.5 micron line is, it's not very transmissive. This goes from 0 to 5% transmission. But we did find these sort of holes, these circular regions in the plant leaf where there was relatively good transmission of this 1.5 micron light. And so I mentioned these holes, which are used for gas exchange, called stomata on plant leaves. We thought these are most likely stomata. It's also known that that these are open stomata where there's greater light transmission. It's known that in grasses like sorghum, stomata grow in rows. And so we thought that these are stomata. This is a figure from that paper also, another indication that this is fairly complicated. All I want to say is we looked at a couple of leaves, we would see these bright spots. And when we started looking, we'd see a lot of bright spots. And by the end of our imaging experiment, we saw less. And so actually, as I mentioned previously, stomata close at night. So stomata were closing during the course of our imaging experiments. So these plants were basically going to sleep while we were watching them. So, we're really now, we can start to image plants where physiologically they're behaving like no light is hitting them at all. So I think this is a pretty involved plot. I didn't want to spend too much time on it. I think this is a simpler system to look at. That these are actually stomata is not only do these go away while we're imaging them, we can pulse light on the plant and reopen these stomata. And so I'm pretty sure we're quite confident we're looking at stoma here. What we'd like to do is, these are sort of static images. We'd like to start getting movies of this process and looking at how stomata may change in terms of different water content or plants under stress or environmental stress or drought conditions. We're working towards getting movies of that. I have no movies to show you right now, at least of stomata, but I do have a movie to show you of simpler targets. So binary test targets, we can image a binary test target in about a second or 2 seconds. So this is a movie of us just moving in manually on a micrometer stage, various binary test targets. So we start this movie with Pac-Man Ghost. We're doing ghost imaging, so we really like imaging ghosts from Pac-Man. So you can see this is a ghost just moved into our field of view, moving side to side. Then, as you may know, in Pac-Man, the hunter ghost can become the hunted. You can start chasing the ghost, and this is sort of a scaredy cat ghost when you're chasing him, a different type of Pac-Man ghost. Next thing, which comes through pretty fast, is a lunging cat. Cats are very popular targets also in any quantum-related image. And then finally, we have a more sedentary cat that we looked at for a little bit longer, that you can see us just sort of moving that cat through the field of view. So I would say this movie was taken with one of these photocathode-based imagers. This was actually the commercial version, LinCam. And so we've been working with this NCAM, we've been working with LinCam. We've also been working with another class of imaging sensors which have a lot of promise in quantum technologies and quantum imaging applications, are SPAT arrays. And so I mentioned we're using a single element, single photon counting avalanche photodiode. People now make arrays of these. We've been working with Eduardo Charbon's lab at EPFL, and he's loaned us, well, not loaned us, I don't know if he ever wants it back, hopefully not, a 32 by 32 element array that they call Piccolo. And, I would say both SPAD arrays and these photocathode-based imagers, they detect single photons. They do it with good timing resolution, and they both can be used for ghost imaging or quantum ghost imaging. In terms of which is better, it's sort of like asking which is better. A pickup truck or a small hatchback. It depends what you want to do with it. I would say, sort of simplifying things, photocathode-based imagers, because they have this great temporal resolution, you can really have this narrow coincidence window, and it's great for these low transmission samples or precise transmission measurements. SPAD arrays, I would say the downside of photocathode-based imagers is they only detect one photon at a time over that whole imaging sensing surface. So they saturate pretty fast at about 1 million counts per second. Whereas a SPAD array, you've got all these individual elements that can do the detection, so it can handle a higher light flux. So I think SPAD arrays are better for faster dynamic imaging at higher light flux. And photocathode imagers are better for these precision measurements on low transmission samples. So I've been talking about quantum light up till now, and we started with this 1.5 micron line because, we were just looking in the literature and thought this would be a reasonable thing to start with. But we've also been thinking about what can we learn from infrared light, classical infrared light, not necessarily quantum infrared light. What in the infrared is a good indicator of plant health and plant response to stress, in particular for these two plants we've been working on, sorghum and camelina. And so we started doing conventional FTIR in reflection mode, single point measurements, not imaging with classical light, to look at sorghum and camelina and how they respond to various environmental stress. So this is FTIR spectra in wave numbers in the bottom and wavelength in the top. I personally prefer wavelength to wave numbers. I've lived most of my life actually doing fluorescence imaging, so I prefer wavelength. But what you can see is under control, cold, drought, salt stress, salt shock, there are various spectral features, which, can be things like the OH stretch, which is in water, as well as polysaccharides. You've got lipid peaks here, 2 and 3. You've got amide 1, protein backbone. You've got various vibrational frequencies, which reflect the macromolecular content of your plant, which also reflect the health of your plant. So I would say, this is just various environmental stressors, what they look like at a plant at a given day. But you can also, do a heat map. This is camelina as a function of time, how these bands change, and can really tell you in time how your plant is doing. This is camelina under drought stress. And so I would say, we're interested in trying quantum imaging at some of these wavelengths, which can tell you more specific, not just water, but other things of interest like lipids and lignocellulose and cell wall polysaccharides. But I want to get your attention on the wavelength of these things. That you're talking 12 microns, 6 microns, 3.5 microns, or 2.9 microns. And so in quantum ghost imaging, you still need to detect that idler photon with single photon sensitivity. And there aren't very many good commercial, I can't think of any really good commercial single photon sensitive detectors, even single element out here. Superconducting nanowires can get you out here, but not the commercial ones. You really need to have the detector be your experiment at that point. But I would say we're interested in going out here, and there might be ways you can do it with ghost imaging. But I think a better way to get out there using quantum-enabled imaging is to use a different imaging technique that Anton Zeilinger developed, as long as Limos and Sven Reimelo and a few others, called quantum imaging with undetected photons. I definitely do not have time to tell you how this works. It does work on you still use entangled photon pairs, and you create potentially two of them, which can interfere, or you don't know what path your photon pair came from. But what's great about this imaging technique is your idler photon in this scheme that you do not even need to detect it. It sort of relies on single photon self-interference. The idler gets absorbed or not, which imprints on the visible pair. So I think what's great about this is you can go further out into the infrared than you can with ghost imaging. But you lose this correlation beat down low light sort of advantage. We are also quite interested in pushing, we did this one wavelength for ghost imaging at 1.5 microns. We'd like to go to other wavelengths, which are sensitive to things like lipids and lignocellulose, which are more important from a biomass accumulation perspective. I think you can do this relatively easily in that, you can just analyze the energy of the photons hitting your bucket detector. And that's, not only now are these two K vectors linked, their energies are also linked because the energy of this photon and that photon summed together have to be the pump laser photon. So you can actually start to do sort of hyperspectral imaging with these techniques, I think, and that could actually potentially be quite useful. Okay, so the take-home message, if you remember anything from this talk, it's that, in quantum ghost imaging, I think it has this really powerful strength that you can probe at one wavelength and detect at another. And the other really useful thing about this is you can do this at a very, very low light flux. We've been using basically light 50 times less than moon-filled light. I would say in terms of expanding this to things beyond plants, I think this is good for label-free imaging of any other light-sensitive sample. Sort of two obvious ones are the human eyeball or anything with a photoreceptor that responds to light. But there are other, materials, things like photoresist, polymers which are light sensitive, things which are sensitive to light. You might be able to image as well by these techniques and have it be more challenging with other higher light flux techniques. Okay, I want to acknowledge Department of Energy, Biological and Environmental Research funded this work. DOE NA22 funded a lot of the RULI and NCAM development, and also DOE BES for SINT. And I'm going to go a little bit over and just want to say that the two people I want to highlight for this talk, in particular, Duncan Ryan. He was a senior postdoc when he started this work. Thanks to this grant, we were able to convert him to a staff scientist, and he's really been the one driving all of this new this microscope development. Katie Seitz is a plant biologist. She's been growing all the plants and doing all the FTIR studies and all sort of the non-quantum plant health studies that I think are important when you add a new imaging technique to a problem. And that's it.
Okay, great. If you have any questions for Jim, please come up to the microphones, which are in the side columns, or ask a question online. Yeah. Yes, please.
Wow, that was just really cool.
Oh, thanks.
You alluded to it. I was wondering about two things. What is the imaging time? So you talked about the time through it looked like it was an hour through the plant sample.
Yes. So for the plant samples, for a 1% transmission sample, I think for the paper we did an hour just to get really good signal to noise. Yeah. But 30 minutes, you need tens of minutes.
Right.
For the plant samples. For the binary targets, it's more like 1 or 2 seconds.
And could you do this in a reflective mode rather than?
Yes, absolutely.
Yeah.
And actually for plants, at these wavelengths, for the IR, yes, absolutely you can. Yes, and we do plan to do that. I just didn't want to, I thought the hyperspectral, saying we're going there, but we also want to go to reflection mode.
Thank you.
So there's one question online which asks, can you do ghost imaging with incoherent light sources?
I would say yes. Incoherent, basically you need something that's, your SPDC conversion efficiency may go down with an incoherent source, but it's chi, you need a material, a high chi2 and the right phase matching conditions to sort of make it a nice environment for these two photon pairs from your pump laser, but your efficiency may go down in terms of the conversion. But once you have the entangled pair, you can still image with it. Would be my response.
Hi, thanks for your talk. It was great. I I have a question about the spatial resolution that you can achieve with this technique. What's the limits of it? What is the current resolution, and what are the limits there?
So there are three things which limit your spatial resolution in these types of quantum imaging methods. One is normal diffraction of your light, lambda f over d type stuff. The other is, just your spatial detector, what's its spatial resolution. But then the thing that really kind of limits you and is sort of different for this is sort of the number of modes of propagation modes in the crystal. It's basically the coherence length of that photon pair divided by the exit output face of your crystal in these near field sort of quantum imaging techniques. And that actually limits you to about 40 by 40 resolution elements. And you can play tricks with magnification, but actually that sort of the number of modes or spatial modes available from the crystal for imaging with these pairs is generally what really, that's the thing of the three terms which contribute to your resolution is the most damning. And you can try to make bigger crystals, but for these highly non-degenerate things, you're using like periodically poled materials, and you cannot make very large area periodically poled materials. When I first heard this was our limit, I'm like, oh, we can just buy a bunch and glue them together. And actually that's not as stupid as it sounds. But there are other things which, if you can work with lower light flux, things like thin films, things like metamaterials, you can use other things which you can create a large XY area, but also, but your phase matching in those materials is not as good as it is in a periodically pulled material.
One more. So for the mid-IR bucket detection, is it feasible practically to have upconversion again for that mid-IR using SFD in a crystal?
Okay, is it possible to have upconversion? I'm sorry, I didn't understand your question.
So you mentioned that the bottleneck is the mid-IR detection and sensitivity for the materials for the bucket detector.
Mm-hmm.
So I'm thinking putting a, let's say, KTP crystal there and upconverting it into visible.
Yeah, yes. Now that's yes. Now I see where you're going. Yeah, that is a possibility. I don't know how much of the temporal resolution you're gonna keep with that sort of a scheme, but that's an interesting suggestion.
All right, thanks.
All right, with that, let's thank Jim again for a great talk. When I talk to my family about the impact of quantum mechanics on the world, one of my main examples is MRIs, and I wanted to make sure we had some ideas of MRIs in this talk. And so I was flipping through Physical Review X, and I saw this amazing paper. And so I'm very happy to invite Evgeniya Kirilina from the Max Planck Institute to talk about her work.
Thank you very much, Ken, for the kind introduction. And it's especially a pleasure to talk in this session. It's a quantum world because MRI is truly based on the quantum phenomenon. And I think it's an amazing success story of a quantum technology because the Zeeman effect, discovered 20 years before even the spin was then identified by Pauli. And it took another 20 years that the spin resonance was detected, first optically, and then in the end of the Second World War in US for nuclei and in the Soviet Union for electrons. And just 20 years later, NMR became the working horse of the analytical chemistry. And then another 20 years later, it was applied for the human body. And I think it's really amazing technology because you can't think about analytics without NMR today and about diagnostic without MRI. And there are much more to come, as you see in this meeting. So just to remind you very, very quickly, in the nuclear magnetic resonance, we use the spins of nuclei. And in MRI, it's in 90% of cases the spins of water protons because it's the most abundant nuclei in the body. In the magnetic field, the energy levels of the spin aligned parallel and anti-parallel to magnetic field become split, and the net magnetic moment of the tissue is formed. And we can induce the transition between this level using the radio frequency pulses, so we can manipulate this net moment and actually look at the spin gymnastic. And if you flip the spin into the plane perpendicular to magnetic field, it starts to precess with a Lamo frequency proportional to the external magnetic field strength, and we can detect the illumination from this radio frequency and use it for imaging. So importantly, the ensemble of spins has a certain properties which are very strongly impacted by the interaction of the water proton with the surrounding tissue. For example, if you think about the longitudinal relaxation rate or time, which is the return of the spin to its equilibrium position, or about the transverse relaxation rate, which is the coherence time of the spins, how they precess in the perpendicular plane, they both are very strongly differ between different tissue. So in the pure water, we would have the relaxation time T2 of about a second, and in the tissue, it's just tens of milliseconds. And that gives us a fantastic opportunity to map, to have this very flexible contrast in the tissue. So one of the main themes in the development of MRI is the steady increase of magnetic field strength. In the conventional standard MRI with a strength of field strength of 1.5 and 3 Tesla, we have a resolution of about 1 millimeter. And this is already fantastic because it allows us to locate the different, for example, the tumors or lesions in the brain, or look at the macroscopic anatomy. With the increase of the fields, for example, the recently clinically approved 7 Tesla MRI, we can look at much smaller structures with a sub-millimeter range, as the cortical layers shown here in this image. But we still can't resolve the fundamental building blocks of the tissue, the cells or fibers. And it's not just a limitation of the technology, so not just a limitation of sensitivity or limited time available for the measurement of the living subject, but it's really the fundamental limit of the water diffusion on the time of the experiment. So for the readout in MRI, we need something like tens of milliseconds. And during this time, the water diffuses a couple of micrometers. So these structures can't be resolved directly. And that's exactly where the physics can help. Because if we would have a perfect biophysical model of how spin interacts with its surrounding, we could actually extract the microscopic information from macroscopically measured spin parameters and push the boundary of what MRI can reveal. So if you wish, we can use the water as a spy, as a probe of the tissue microstructure. And water is involved in multiple interaction with the tissue at very different temporal and spatial scales. For example, in order to achieve the longitudinal relaxation, the spin has to give up the energy, and that's the most efficient for the very fast fluctuation with the times in the order of inverse Lamo frequency. And this appeared to be molecular rotation. So, for example, macromolecules, so slowly rotating macromolecules, are really inducing the T1 relaxation, and then we can infer a macromolecular tissue component from the T1. And I am very interested in the T2 and T2 star because they give us the information about the microscopic magnetic field fluctuation in the tissue. So magnetic microstructure within the tissue, for example, the presence of the metal, paramagnetic metal in the tissue. So in order to harvest this information, we go in the 3-step approach. The first 2-step approach, actually. We first use the physical model to convert weighted images recorded in MRI into the quantitative maps of spin parameters, such as relaxation rates. And then in the next step, using the biophysical model, we link these quantitative parameters to the microstructural histological features of the tissue, such as cell density, cell sizes, myelin concentration, or cellular metals. And you can imagine that creating this model is extremely difficult because even in the tiny small voxel in the human brain, it has a very, very complex composition. It contains thousands of neurons, a lot of fibers, glial cells, and vessels. So we have to reduce this complexity in order to infer some information. But it turned out that in terms of magnetic properties, the 2 major components which drive the contrast are actually diamagnetic myelin, which is the isolating substance around the axons, and paramagnetic iron. So the goal of my group is to develop the new method for map iron and metal with cellular sensitivity using MRI. When you first hear about the iron in the brain, you may wonder, and you may ask how much iron does the brain contain actually. And it's not much, so it's 200 milligram. So it's like two tiny screws you have in your glasses. But if you compare it with the biological tissue, for example, spinach, it's actually as much as in the 10 kilogram of spinach. So it's a very, very iron-rich organ, actually the second iron-rich organ after liver. So why is it the case? The point is that iron is a highly catalytical metal, which can catalyze a lot of useful reaction and is used by the body for oxygen transfer, neurotransmitter synthesis, and energy production in iron-sulfur cluster. Therefore, brain really needs iron. However, iron is a double-edged sword, and when it's present in a too high concentration, this high catalytical activity turns evil, can reduce oxidative stress, and actually in all neurodegenerative diseases, we observe high concentration of iron. So non-invasive method to monitor brain iron are extremely highly needed, and MRI can be such a method. So for the rest of my talk, I want to show you one particular application, because I think it's a very nice illustration how it may work. And this is to map one very specific but very important type of cells in the human brain, the dopaminergic neurons, which are very important in context of Parkinson's disease. And what I will show you, it's actually a PhD thesis of one very talented student in my group, Malte Bramelow. So you all know that the Parkinson's disease is the second most prevalent neurodegenerative disorder affecting many, many millions of people on the earth. And it has a very bad motor symptoms, rigidity and tremor, which make the quality of life very bad. And the reason for the symptoms has been discovered already 100 years ago in Sorbonne, and that's neurodegeneration of very specific neurons located in a very tiny nuclei, just a bean-sized nuclei, substantia nigra. These neurons are very special. They are arranged in the small cell clouds called nigrosomes, shown here in the red color. They are pigmented and they are very iron-rich. And interestingly, the tragic thing is that at the moment when the first motor symptoms occur, already 75% of the cells is lost. So actually the curative treatment for Parkinson is not possible because the decade before, when all the cell dies, we just don't realize that. So if there would be a method to detect this loss, that would be a game changer. So let me explain a little bit why these cells have a lot of iron and why is it actually the window of opportunity to detect them with MRI. The reason is that iron is needed for dopamine synthesis. It is catalyzed by the enzyme tyrosine hydroxylase with the 4 iron atoms. And this is how the dopamine is produced. But on the other hand, when the dopamine reacts with the dissolved iron in the cells, it itself converts in the very toxic quinone and can produce oxidative species. So the cells have to deal with this really poison synthesis, and they kind of make a waste bin, which is the pigment neuromelanin, which is a dark pigment. So this structure becomes really dark. It's similar to the pigment we have in our skin, actually. So this is why it's also called substantia nigra. The neuromelanin is a sponge which can have 2 different sites for iron storage. The one is the ferrohydride crystal with a with a high binding site and very high affinity, so very safely. It is antiferromagnetically coupled but behaves as a paramagnetic at body temperature. And the second binding site is the single atomic binding site with the paramagnetic property, which seems to has a much lower affinity, so be more neurotoxic. So it's not clear how exactly the iron is distributed between those, but the idea now is that in the course of life, the neuromelanin accumulate, the iron accumulate, and at a certain point, these things got overloaded and the cell die. So if we would have an ability to detect the certain, critical level, that would open the possibility for treatment. So that's a strategy in my group, how we try to achieve this. We combine ultra-high resolution MRI in vivo with the postmortem histology on the postmortem donated tissue, where we can also do the histological processing and find the ground truth about iron distribution in cells, create biophysical model of MRI contrast, validate it, and then apply it in vivo. And I will walk you through the different stages. So let's first try to understand how the MRI of the single neuron would look like. And it's almost a perfect model for physicists because you can approximate it with a magnetic sphere containing certain amount of iron. And we all know that in magnetic field, there would be this beautiful dipole field formed around the cell, and the magnetic moment will be proportional to the susceptibility of iron, magnetic field, and the total amount of iron. And now the water molecules sitting close to the pole wouldn't experience stronger magnetic field than those sitting around the equator, and this would cause a dephasing. And interestingly, we can now use to invert this equation and determine susceptibility if we would know the magnetic moments and the mass of the iron. And that's exactly what we do. We combine MRI microscopy to determine magnetic moment of the single cells, and we use the proton-induced X-ray emission and X-ray fluorescence to measure the iron mass. So on the next slide, you see the image MRI microscopy of the tiny block of postmortem tissue from substantia nigra with black dots. And these black dots are actually dopaminergic neurons. And they are black, they are dark because the spins are dephased around those cells. And on the bottom line in color, you see the phase of the signal. And if you try to, so it's actually exactly this dipole field I have shown, but very bad resolution. And this is interesting because here the situation is inverted. In normal MRI, we would have a big object. We would have a voxel or pixels, which are much smaller than the object side, and the diffusion length is much smaller than the voxel. And here everything is other way around. So diffusion length is actually similar to the cell size or the object we're interested in and comparable to the voxel size. So we need a little bit of modeling to deal with this problem. And we use the super resolution approach. So we simulated the signal, as you see on the right side, and fitted the signal how it should be to many different neurons. So we determined their magnetic moment. We then performed the histological processing of the tissue and went to the synchrotron in DAISY, where you can use the fantastic sensitivity of synchrotron and the very high resolution map iron in the cells quantitatively. And then the big work was to align, this histology and the MRI, which seems easy, but wasn't actually, but might have managed. And now we can plot the magnetic moment of the cells as a function of the iron load. And you see that they all lie on the very nice line. So it's now the cells of two different donors, and we can fit the line to it and determine susceptibility of iron bound into the neuromelanin. And I think that's very exciting because the susceptibility was measured directly in the cell without any prior isolation, which is very important because it is known that any treatment, any isolation from the tissue change the binding sites of metal. It has a very high sensitivity, so we can detect picogram of iron per cell and work on the large cell population at room temperature and physiological conditions. And this is particularly important because if you want to translate it to MRI, we really need big cell ensembles and conditions. So we are doing very well compared to other methods, especially because we are working at room temperature, and even our main competitor, the nitrogen vacancy magnetometry, is working in 2D slices, so we are still kind of performing better. Now let's look on the value we obtained. It's actually also very interesting because now comparing it with what I told you before about the neurotoxic and neurobinding side, we can see that the susceptibility is much close to the neurotoxic binding sites, which is probably the dominated form of iron in neuromelanin. So this actually may explain the iron contribution to vulnerability of these special cells in Parkinson pathology. Well, so now we come from the single neuron to a single voxel in the brain, so make a next step. And now the things become more complicated because in the one voxel we have hundreds of cells. We have to average their phase of all spins, and it can't be solved analytically that simple. However, there have been a beautiful theory developed by Dmitry Yablonsky and Mark Hecke in the 90s, where they have shown that for the one very specific case, if the magnetic field perturbation induces by localized magnetic inclusions, we have a nice analytical expression for long-term regime. And the decay is just proportional to the product of magnetic susceptibility, magnetic field, the total mass in the inclusion, and their volume fraction. And this is a very nice because we can now invert this equation, and by dividing the measured relaxation rate, which is measured macroscopically, by the susceptibility value we just determined in the previous experiment, we can obtain biologically interpretable value, the product of cell density, so their volume fraction, and their iron load. And that's very relevant. So now we validated if the theory really works. So we created, using the synchrotron data, we created the one map of the one voxel, so about 0.5 millimeter size. We convolved the iron concentration we measure with the dipole, created the local field, shown here in the red, local field perturbation within the one voxel, and then used the theory, shown here in red, Monte Carlo simulation, which account for diffusion, and compared it with experiment. And all of that actually merged very nicely. So the theory can be used, and we really can interpret the relaxation rate measured macroscopically. So actually here I could stop my talk because actually the physics would be over, right? We solved the problem. But I want to show you some other steps which shows what does it need now to take it further to real clinical translation. So now we come from the single voxel to the brain nuclei. So we go to the domain of radiology. And when we started first to look at the literature, radiological literature, we were kind of shocked because our wonderful theory, which was validated so well, contradicted the big body of the clinical literature. So in the radiology, there was the one particular sign called swallow's tail sign, shown here on the left, which is the bright part of the substantia nigra, and this was associated with the nigrosome 1. Because of two reasons. First, it disappeared in Parkinson's disease, and second, there was the one study which compared histology and MRI in two-dimensional slices and showed they kind of co-aligned. So this is a fundamental contradiction between the common view that the nigrosome should be bright and the prediction of our theory showing that neurons induced relaxation, it should be dark. So as a physicist, you always believe in theory and never believe in the anatomy. So we were puzzled by the point and realized that it's actually a very challenging point because the structures we are talking about, they're really, really tiny. So the substantia nigra is the size of the bean, and the nigrosome are just 0.5 millimeter thin. So if you co-align histology and MRI, it's really easy to make a mistake. So we decided to repeat an experiment in a different way using the 3D co-alignment. And here we luckily has a fantastic collaboration with a group of Birte Forstmann in Amsterdam and Annike Alkemade, who created the 3D multimodal data set on brain structure on the postmortem brain. And we were lucky to be able to measure them with MRI before they went to the histology. So this is a fully 3D data set. We have all the MRI contrast, all MRI parameters, and then multimodal histochemistry. So, and that was actually, this work was done during the time of pandemic where we can't go to the lab, but we could do a lot of coloring on the screen. And what we have done, we created this histological atlas of nigrosomes, which lived in the three-dimensional space and was also co-aligned in 3D with MRI. And after we compared the delineation of the swallowtail shown here in the blue and the nigrosomes in the histology shown here in the red, we have seen that they overlay, but only partly, and actually has a very different shape. So also they overlap, they actually, partly and come together in the same place, but they're actually not the same, just as a bread and a hot dog. So I don't show it to you to learn you about the neuroanatomy, but I think it's a very nice example showing that understanding of the contrast mechanism as MRI can really help you and can really guide understanding of, or even for radiologists. Right, so now after we get the theory right, we know where to look for those cells, or for the nigrosome, we go into the direction of clinical translation. And here the most important point is that the structures we look at are really, really small. So it's really difficult to resolve them. And here I want to show you the work of my colleague Ekkehard Peik, also in Leipzig, who developed a method where we can obtain quantitative maps of quantitative parameters with a 0.5 millimeter resolution. And so actually this is exactly the resolution we need to be able to see the nigrosome. So now we are very well equipped. So that's the last puzzling point in this work. And at the moment, as we speak, this method is applied in the big study across European Union with the several sites in Belgium, France, and Germany, where we look at the patient cohort of the healthy people with the risk of Parkinson and de novo Parkinson patient, and we plan to scan them with several years, to follow them by several years, and to look if we really can see the neurodegeneration of the small nuclei in order to be able to detect early stage, detect the biomarkers, so to create early stage detection, and also to understand the role of iron in Parkinson's disease. And with that, I would like to come to summary. I hope to show you the new sensitive method for in-cell magnetometry of paramagnetic metal, which can not only be applied to dopaminergic neurons, but actually to many other cases. So we are also, that was just one example, we are also working on the iron-rich plaques in Alzheimer's disease or on macrophages in the context of inflammatory disorders. I also hope to show you that the macroscopic relaxation rate parameters are sensitive to the cellular distribution and chemical form of iron binding, and that the very specific information about the brain iron can be obtained by combining ultra-high field MRI with biophysical modeling and advanced histology. And I think the new generation of diagnostic method with cellular specificity can be achieved with this work. And with that, I would like to thank my colleagues in Leipzig and especially Malte Bramelow, whom I mentioned, and Nick Weiskop, who is leading the Department of Neurophysics, which is the great department, the fantastic team, and a lot of colleagues in different institutions and across different disciplines. And thank you very, very much for your attention.
Plenty of time for questions, and please come up to the microphones.
I want to know, is there any correlation between the Parkinson's disease and the average level of iron in the blood of people?
It's not that simple. So the brain actually regulates iron very, very carefully. So there are import through the transferrin receptor and the very careful management. So there is no direct correlation between your plasma iron levels and your brain iron levels. I think in the case of iron deficiency, you would have also you may get the low brain iron, but it's not necessarily directly correlated.
Thank you for the very inspiring talk. You mentioned it, but of course, hemoglobin is a massive source of a massive load of iron in the brain, and I'm wondering, one, does the iron in hemoglobin itself interfere with your ability to resolve your measurements? But two, would there actually be ways of using the hemoglobin and the iron in hemoglobin itself as another metric of brain disease or change in brain function?
Right. So you're absolutely right that, of course, the blood in the vessels is a very strong contrast source. And we are very lucky because due to the fact that there is the functional magnetic resonance imaging, the people know a lot about it. So in functional magnetic resonance imaging, you exactly look at the effect of the blood. So they're very good biophysical models. And it doesn't contribute too much to the contrast. So there is a certain contribution, actually in the work of Dmitry Yablonsky, he have shown that you can also kind of remove this part because it has a quadratic decay at the very early times. So it doesn't seem to contribute too much, and it's a more or less constant background across the brain region. And concerning your second question, of course, for example, the interesting thing is there is a blood-brain barrier, so the iron from hemoglobin doesn't directly leak to the tissue. And if that happens, for example, the context of Alzheimer's disease, then you see a lot of contrast changes there. For example, in the context of development of the new treatment against Alzheimer, the one complication is exactly the breakage of the blood-brain barrier, and it just comes with so-called abnormality in imaging. So you start to see it, you start to see a really iron rim. So we would see microbleeding, and that's become important in certain diseases, but not that much in Parkinson.
Any more questions? Nothing's online. Actually, I was really impressed by the wide range of techniques you had to pull together to actually get this to work. So, can you talk a little bit more about how when you use the X-ray fluorescence to kind of get the voxel size into a shape to figure out what the magnetic pieces are?
Yeah. So for the X-ray fluorescence, so what we need for our models, we need the we need the maps of iron with the cellular distribution, so with the resolution smaller than the diffusion length of water, so a micrometer. So that's why the X-ray fluorescence is important. But on the other hand, we also need the large coverage because MRI is always looking at the ensemble. So we'll look at the macroscopic volumes. So this is why XRF is actually one of the unique methods to do so. So how we do it practically, we use the postmortem tissue, which is then embedded, cut in the slices, and then we use their XRF. There are fantastic beamline at Daisy in Hamburg, where we can quantify different elements. It's not just an iron; it's actually much more. It's also all different kind of elements we can get there. Yeah.
All right, let's thank Evgeniya again. So the other thing after MRI that I use to talk to my family about quantum mechanics, I usually talk to them about GPS and atomic clocks. But then I wanted to have a connection somehow with atomic clocks and the April side of the meeting. And then Ekkehard Peik had this wonderful paper on nuclear transitions, which could potentially become amazing nuclear clocks. So let's welcome Ekkehard Peik.
Yeah, it's my great pleasure to add an additional quantum system to the topics of this session. And it is from a field that's the interplay between quantum optics and nuclear physics. So I will report about the nuclear laser excitation and work motivated by developing a nuclear clock. So if you Google nuclear clock, you may find different topics. I'd like to briefly exclude what I'm not going to talk about. There's, of course, radiometric dating, so measuring time in archaeology or in geophysics based on the decay of an isotope with a known half-life. Of course, a very powerful and method, but we are speaking about timekeeping in a different sense. And of course, also, I'm not going to speak about the nuclear doomsday clock. Rather, I would like to introduce the nuclear clock, more or less following the concepts that are well known and from the atomic clock that we use today, like the cesium clock. The frequency of the clock is always determined by a quantum two-level system in an atom, in a molecule, a system with two preferentially long-lived energy states, energy levels, and their transition energy is related to a transition frequency. The resonance is usually driven with an oscillator. In the cesium clock, it's a microwave oscillator. In the optical clock, it's a stable laser. This laser is used to probe the absorption of the response of this system. There is a state detection, a servo system that steers back the system of the oscillator to be in resonance with the quantum mechanical two-level system. And then in order to establish a time scale, one needs a clockwork. Of course, microwave can be counted easily electronically in the optical range. One needs a femtosecond frequency comb in order to down-convert the optical frequency into the countable range. And now this is well established, and all the atomic clocks that we know today rely on resonances in the electron shell of an atom. So there is processes related to a valence electron. And the nuclear clock I'm going to introduce is making use rather of an electromagnetic transition inside the nucleus, not in the electron shell, but inside the nucleus. What is the advantages, or what can be gained from this? It's mainly 3 points. It promises a high accuracy because of the absence of systematic shifts. It promises high stability because one can do it not only in the very dilute gases, but also in a solid state medium. And it promises high sensitivity to new physics for clock-based searches for new physics. And I'm going to explain these 3 advantages in a little bit more detail. Why, given these advantages, why has it not been done 50 years ago? Why is no nuclear clocks established already? We need to look a little bit at how do atoms and nuclei interact with electromagnetic radiation, or especially with light. And there is quite some substantial differences. So for atoms interacting with light, of course, we know there is 2 signs of charges. There is the electron and the proton, binding energies in the electron volt range. For nuclei, there is only 1 sign of charge, and the binding energies are usually much higher in the range outside of the optical spectroscopy. The electron bound and the atom bound by a central force that determines the symmetry of the wave functions and the selection rules for the electromagnetic transitions. Situation in the nucleus more complicated. We have tensor forces, spin-dependent forces. So already the simplest bound nuclear system, a neutron and a proton, the deuteron already is no longer spherical symmetric. It has an electric quadrupole moment. And concerning the strengths of the interaction, magnetic and electric interactions, we also see different scalings. For magnetic radiation, of course, we know in the atom it's determined by the Bohr magneton. For the inside the nucleus, we need to scale from the electron mass to the proton mass. So it's the nuclear magneton about a factor of 2,000 smaller. The second, for electronic interactions, of course, you know there is the criterion that the antenna for emission or absorption of an electric wave usually has to be in the range of the wavelengths in order to be efficient. And for an atom already, this criterion in the optical range is not well fulfilled. So the R over lambda in the optical range may be 10 to the minus 4, the nucleus being much smaller, this antenna factor, of course, several orders of magnitude smaller. So it means that we should expect electromagnetic transitions in the nuclei to be significantly weaker than we are used to from atoms. So for magnetic dipole transitions, it scales with mu square, so about a factor 10 to the 6 suppressed relative to the atomic scale. And for the electric multiple radiation, it is the R over lambda factor to the power 2L. So for the dipole, again, factor 1 million. So we need to expect these processes to be relatively weak. And then in addition, there is also, I mentioned on the top, the energy mismatch of the 1 MeV. Well, this is not a unique criterion. There are some nuclides that have lower transition energies. If you look here at the chart of the nuclides, what is shown here is the energy of the first excited state. The color coding shows red, the high energies, and green, the low energies. You can see the magic numbers, the shell closures, where there is a very deeply bound ground state, and then the energy of the first excited state is also relatively high. So in order to find lower transition energies, one has to stay away from these regions. And you can see the green regions, especially in the heavy nuclei, like in the actinides. But still, the green scaling here is 20 keV, so still a relatively high energy compared to laser technology. But the one exception that I will focus on is the thorium-229 isotope. It was studied in gamma spectroscopy in the mid-1970s already, and the level scheme as shown here was established. You can see the rotational bands that are typical for nuclei in this Mars region. And then you can see that the ground state is one band head, and then there is another band head of a different rotational band, and it's very close. Here the energy is marked as about 4 electron volts. And the transition between these two levels is the one that we are interested in. The nuclear structure in this domain is described by the Nielsen model. So it describes a collective motion of an elliptic core and then an unpaired nucleon, in this case, in the case of thorium-229, an unpaired neutron acting like a valence nucleon, and it may take different orbits and different spin orientations and coupled then to the core containing all the other nucleons. A very powerful model that can predict quite something about the structure, but it's not a complete description. So, for example, in the case of this thorium-229 transition, one would expect, if it's really just related to the neutron, that the charge distribution, the proton distribution does not change. But in doing hyperfine spectroscopy already a few years ago, we measured that the RMS charge radius of the ground state and of the isomer is not the same, but it's different by this quantity given here. There is not really a microscopic picture of how this very low energy appears in this nucleus. But a very interesting aspect has been pointed out by Victor Flambaum several years ago. And he was explaining that this very near degeneracy of these levels must be due to some kind of magic cancellation. There is contributions from the Coulomb energy, from the strong interactions inside the nucleus. And by relatively simple estimates, one can see that they should change on the scale of 100 keV, maybe a mega electron volt. And so in order for the sum of the energies to come out at nearly the same value, it means that in exciting the neutron, a change in the Coulomb energy must be compensated by a nearly the same magnitude but opposite same change of the strong interactions. And this means that the system is so highly sensitive to tests of fundamental physics, because if you think, for example, about searching for variations in the strength of the fundamental interactions, if there would be some change in the coupling of electromagnetic and strong forces, it would have a strong influence on this very delicate balance here. And that established the high sensitivity of this nuclear clock for tests of fundamental physics. I'd like to come back briefly to the other two aspects of the high accuracy and the high stability. The high accuracy is that the nucleus is somewhat protected from the environment by the electron shell. Collisions, for example, but also external electric fields are somewhat first interacting with the electrons and not so much with the nucleus. And the core idea behind the nuclear clock, behind the accuracy, is that one is a choice of the suitable electronic environment. So we are mainly interested in the nuclear transition, but we will always work in some electronic environment that we can select. And now we can make a good choice for high accuracy, for low sensitivity, and one can make a less good choice. And this was an aspect of the proposal. So we first pointed out that it was the spherically symmetric electron configurations that are especially good for the clock. And later, Campbell, Derivianco pointed out that it was also be the stretch states, the states where electronic and nuclear angular momentum would be maximally aligned, and these would be the ones where systematic shifts would be optimally suppressed. The high stability of the nuclear clock would result from the fact that the nuclear transition, the gamma spectroscopy, one could also do at very high resolution inside a solid density. And this, of course, opens the perspective not to work just with a single atom, maybe as in a single ion optical clock, or with a dilute ultra cold gas in the optical clock, but rather at a solid state density. So increasing by an enormous number the amount of particles that can be probed for the clock, and of course, leading to a much higher signal-to-noise ratio. It was also investigated and analyzed in different systems, and two groups, especially at UCLA and at TU Vienna, started to grow doped crystals for this purpose. So I'm switching to the experimental part, going to the status of knowledge about this nucleus about 2020. This was when we obtained an ERC grant for this interdisciplinary research, and the plot here shows the nuclear two-level system now, the ground state, the excited state, and all the numbers as they were known at the time. And I'd like to focus your attention especially on the transition energy, at the time 8.2 electron volts, or converted to wavelengths 150 nanometers, but in the vacuum ultraviolet range. But you can still see with a significant uncertainty of about 2 nanometers at the time. But we thought, well, this information was good enough for us to start building a dedicated laser system. So we needed to build a laser system that would produce this VUV radiation about 150 nanometers, and that would be sufficiently tunable to cover this uncertainty range of about 1 or 2 nanometers. So we decided to use 4-wave mixing, a well-established method in laser generation. So basically it is mixing 3 photons inside atomic xenon gas. 2 ultraviolet photons, about 250 nanometers, are tuned close to a 2-photon resonance in xenon. Xenon has a number of suitable resonances at 8, 9, 10 electron volt. These 2 photons create a polarizability inside the xenon, and then we shine in an additional third infrared photon, and then the sum of the 2 ultraviolet minus the infrared photon energy is emitted as coherent ultraviolet radiation. It's a high-order nonlinear process, so it's not very efficient. One needs intense fields, so we need to work with pulsed amplifiers starting from CW radiation that we use for control of the frequency, but then we need to go to pulsed dye amplifiers to produce nanosecond pulses of a few tens of millijoules energy. These are focused into a xenon cell, and finding the right pressure, the right angles between the beams for phase matching, one can obtain then pulses of a few 10 microjoules energy in the VUE. Then, of course, one needs to work in a vacuum beamline, shown here coupled to an ion trap. When we had this system running, we looked at the parameters, what would be the excitation rate that we would obtain from the nuclear transition, and it turned out to be disappointingly low. So we decided not to do an experiment with trapped ions. The signal would be too weak, but rather go towards the doped crystal. And it was just right that the Vienna team at the time had made great progress in the development of the doped calcium fluoride crystal. And we decided to use this system for the search for the nuclear transition. So the experiment is shown here. So we are producing the light in this 4-wave mixing, as I have explained, and then we would drive the nuclear transition, and the nucleus would be embedded in the calcium fluoride. The calcium fluoride band gap is significantly bigger than the nuclear excitation energy, so we would expect the nuclei to be able to interact with this VUV light without much interaction with the electronic system of the host crystal. The thorium is embedded in the 4+ charge state, replacing a calcium, but the calcium would normally be a 2+ ion, so 2 fluorine F- ions would need to accompany each thorium in order for charge compensation. The picture on the right shows a photograph of the crystal. It's a few cubic millimeters containing like 10 to the 16, 17 nuclei. Just for the purpose of the picture, we illuminated with a blue laser. It is, of course, a colorless crystal of optical quality. And now I'm skipping a lot of experimental efforts and challenges that we needed to overcome, difficulties with the experiment, and still searching this range. But after this, at some stage, we found a signal. On top, you see more or less the raw data changing the frequency of the laser. We were expecting, of course, the like several minutes lifetime of the excited nuclear state. So we would shine in the laser for like 2 minutes and switch the laser off and detect in the dark the expected nuclear fluorescence decay, in this case for 2.5 minutes. And this, although you may say it's a slow scanning, but based on the lifetime of the excited state, it's rather fast. And so we see a characteristic asymmetric line shape. The red points increasing the frequencies from low frequencies to higher. We see a relatively rapid increase in the fluorescence signal and then a slower decrease, which is just due to the slow decay of the nuclear excited population that we have created maybe already in the previous laser pulse. So this is easily understood. If we reverse the scan direction in the top graph, so for the blue curve, we just get a mirror image of this asymmetric line shape. So once we know the lifetime, we can easily do some post-processing of this effect, and then the two lines nicely overlap, and we get a line shape with a 10-20 gigahertz line width as determined from the frequency resolution of the 4-wave mixing pulse laser system. And then at this stage, we were, of course, happy and excited, but we knew we had to be very careful, because what are we doing? We are shining high intensity, high energy VUV photons on a solid state. So we may do charge separation, we may produce color centers, there may be some impurities, maybe some rare earth ion with a long-lived metastable state. And so we had to make sure that what we are observing was really the nuclear signature and not some other electronic parasitic effects. And we did a number of test experiments. The most important one is shown here in the lower graphs. This is a control experiment with a crystal produced with the same methods in the same laboratory, again in Vienna, but now with the naturally abundant thorium-232 isotope. So it has the same chemistry, the same crystal growth procedure, but of course a different nuclear, same electronic structure, but of course a different nuclear structure. And so if we expected a nuclear signature related to the 229 isotope, it should be absent in the 232. And indeed that was the case. So at that stage, we were pretty convinced that what we had seen was really the search for nuclear clock resonance. We wrote a paper, we submitted, we did some more measurements. You can see here data we took later with a bit more averaging and signal to noise. We used differently doped crystals with different concentrations, showing different signal strengths. Line shape, again, always determined by the laser system, but we also find consistent frequencies for the differently prepared crystals. We looked at the fluorescence decay curve, so there's a lifetime we could measure precisely, of course, just following the decay after switching off the laser. And the blue points you see here are just the experimental raw data, and the black solid line is just a fit to a single exponential. And this is really like you would expect from the textbook of nuclear physics. It's a very simple single exponential nuclear exponential decay. And we checked that the decay constant, the half-life, so to speak, would also be independent from different parameters like the doping concentration. One of these crystals underwent a special chemical treatment, a refluorination. The calcium fluoride tends to lose fluorine, which modifies the optical properties. It can be brought under a CF4 atmosphere, and then it can be refluorinated. the same lifetime. We changed temperature from about liquid nitrogen to above room temperature, and we always found the same lifetime as expected for the nuclear process. And then also after announcing our results, we very soon learned from results from the group of Eric Hudson at UCLA, who was working with the same nuclear system, with a similar laser system, but with a different host crystal, LISEF, and he found the resonances at exactly the same frequency to within the gigahertz resolution, and again confirming that what we are looking here is really the nuclear process. If it would be any electronic resonance changing the doping, the host crystal would shift the resonance by maybe gigahertz or terahertz. The plot on the right shows the historic evolution of the information about the nuclear transition energy. So the blue points are the one from the pre-laser area based on gamma spectroscopy. You can see the last one published in 2020 was especially important for our search. This was from an experiment done at CERN from the Isolde apparatus. And then you see the about factor 1,000 improvement in the accuracy and the nice from the laser experiments, and the nice agreement between the PDB and UCLA measurements. And then in late summer last year, there was a third report now making significant progress in the resolution. The group at Gila in Boulder, again using crystals from Vienna on calcium fluoride, actually from the same crystal growth batch that we had also used in Braunschweig. They used a different laser system, so the VUV radiation was generated from a femtosecond pulsed laser system, a frequency comb stabilized in the infrared, and then in a single stage harmonic generation. Again, xenon was done to transport a part of this frequency comb in the vacuum ultraviolet. And then there's a comb of all these narrow lines, and then now an individual line can be tuned across the nuclear resonances. And they found like a sub 100 kilohertz resolution, and obtaining a kilohertz with a kilohertz range uncertainty for the transition frequency. And you can see it really opens now a field of studies, of precision studies for this, for the thorium doped in the crystals, and also enabling first tests of the system as a clock. I'd like to quickly shift gears a little bit away from the solid state systems, because from the beginning we had thought that the high accuracy system may actually be with trapped ions, thorium ions in an ion trap. Here the methods would be different. We would not have this large number of particles, and so we could not simply look at the fluorescence, one photon every 10 minutes or so. But we would have another very powerful detection scheme based on a quantum effect like a nuclear electronic double resonance. And this is explained here. So we would have, again, some electronic environment. We would use a laser to drive an electronic resonance in the electron shell. And in the left picture, you can see here's the nucleus in the ground state. We would observe fluorescence from the shell. Then we would drive the nuclear transition, excite the nucleus. It would change the spin. It would change the magnetic moment. So the hyperfine pattern would change, and we can easily read this out on the electronic transitions, being able to scatter millions, billions of photons for a single excitation of the nucleus. So we would have a large quantum amplification like Demel's electron shelving that is used in the optical clocks with single ions. And in fact, a few years ago, in an experiment, we had already shown that this hyperfine detection of the isomer is possible. So this is an experiment where the isomer was produced in the alpha decay of uranium, and then an ensemble of thorium 2+ ions in this case is trapped. Most of them, 98%, are in the nuclear ground state, but 2% are in the excited state. And we were able to distinguish the hyperfine patterns of these two ensembles. The strong resonances that you can see here are from the nuclear ground state, and the ones with the cyan color are the resonances of the isomeric state. And this measurement also allowed us the very first measurements of the nuclear moment, the charge radius, and the quadrupole moment of the isomeric state. And so this will now be used as a detection method in a trapped ion clock. We are not yet there, but we are preparing this experiment to trying to excite the nuclear resonance in the trapped ions. I show you some pictures here of an ensemble of trapped and sympathetically laser-cooled thorium-3+ ions. So we are trapping them together with strontium+ ions. Strontium+, thorium-3+, they have a similar charge to mass ratio. So in a radio frequency Paul trap, they are quite closely coupled. And if we laser cool the strontium+, which is very convenient and easy, this is sympathetically also cooling the thorium-3+. In the picture you see here, the fluorescence only of the strontium is detected. So the thorium-3+ that are embedded in these crystals appear as dark spaces, somewhat bigger than the space occupied by strontium, because of this 3+ charge. So in the middle of these huge cigar-shaped configurations, you can see a linear chain of maybe 10 to 15 thorium ions, and the bottom picture shows like 20, 30 thorium 3+ ions in a now three-dimensional structure. And we recently used these structures to perform high-resolution laser spectroscopy, in this case of an electronic resonance. Here we are showing a completely resolved hyperfine pattern of this 690 nanometer transition in thorium 3+, and this is all the ground state. And we are using this for improving the knowledge on the nuclear structure, on the nuclear moments. But I think we are also now prepared to use this system for the laser excitation of the isomer. So I'd like to give a little bit of perspective now. Where are we with the clocks? This graph here shows the Moore's law, the logarithmic, the exponential improvement in the uncertainty of clocks. The black dots are the cesium clocks, starting from the very first one built in 1950, and then improving the accuracy and now leveling off a little bit in the low 10⁻16 range. The red points are from the optical clocks, starting like 30 years, 40 years later, but then at an improved slope, at faster progress, and also reaching now about a factor 100 lower uncertainty, with all the optical clocks maybe also showing a little first indications of leveling off. And now, don't take me too seriously, but I could not resist adding the information here on the nuclear transition energy. You can see we are still, we started from many orders of magnitude above the state of the art in the clock. We are still several orders of magnitude above, but you can see that the slope in the present year has been pretty impressive, and I think it gives us hope that we will see good progress in this field and interesting developments. So this brings me to the conclusion. What's next? So all the experiments, the 3 experiments that I have reported, they all made it to the cover of their respective journals. They were all limited, but by the line widths of the laser systems used at present. So there are some now that we know the target wavelengths, people will develop narrowband VUV laser sources in order to benefit from a much higher frequency resolution. There's now a lot of studies going on. There have been some presentations in this conference on dynamics of the excitations in the crystals, on the line shapes. New materials are being investigated. Thorium fluoride has been investigated as the first stoichiometric crystal, where thorium is really not a dopant, but part of the crystal lattice. And as I mentioned, we are very interested now in exciting also the transition in the trapped ions, where we obtain a still better control of systematic shifts. And then I think it's not going to revolutionize GPS system within the next 5 years or so, but I think the interest is really to have this clock and to compare it to the established optical clocks in tests of fundamental physics. I think this will be the very first scientific application. Because of the high sensitivity, we can benefit from using this nuclear clock already, even if we are not yet competitive with the absolute state of frontier, state of the art, and the optical clocks. So I think there is much more to be expected in the near future from this field, and I'm looking forward to this development. I'd like to close by thanking the team, the wonderful cooperation that we had between Braunschweig and Vienna. So there is my team in Braunschweig and the team of Thorsten Schumm in Vienna. We happened to meet last year and to take this picture with all the authors in front of Technical University of Vienna. I'd like to close and thank you for your attention.
Question from John Doyle.
So it's an amazing scientific accomplishment. Really, it's incredible. And first, I'd like to thank you for getting it done on time for the International Year of Quantum Science and Technology. It's a great story to tell, and there's only one part in my mind that's missing. There's the scientific applications, seem clear, and I can explain those to the public. What are the dream technological applications? So if that curve continues down and we have a 10 to the minus 22 clock that we can put on our wrist, what are you thinking, dreaming very broadly, could be done?
I think the most important questions are really the questions from scientific applications like testing the foundations of relativity, of quantum theory. So you mentioned so in principle, if you may dream of a clock at 10 to the minus 20 or so uncertainty, we could do very precise local measurements, of course, but as soon as we would try to establish a time scale that is transfer time to a different city, to a different continent, of course, we would need to take the relativistic effects into account. So this would be like a technical challenge for practical applications, but it would be a great laboratory for testing relativity. And this, I think, this is now the main obvious development that I would think about. I'm also thinking more about other applications beyond the clocks, like looking into nuclear physics. I think, establishing a new tool for doing precision measurements of nuclear physics, just on a very specific system, but with an unprecedented accuracy. And then I think maybe again the clocks are a little bit ahead of the applications, like the cesium clocks were. When they were first built, the practical application of GPS was not seen, but the clocks were there, and then people had this idea, oh wow, there's these fantastic clocks, now we may use them in navigation. And maybe something groundbreaking ideas may come again from having these improved clocks available.
Great, thank you.
So I know oh yeah, go ahead. I'll just ask my question real fast, Ekkehard. So one scientific application is always this question of, is there like a time variance of the mass of the electron versus the mass of the nucleus? And it seems like in this, comparing this nuclear clock versus atomic clocks should be super good for that. Do you have a sense of how much better it could improve the balance?
The sensitivity factor for this kinds of tests is still partly an open question. What was mainly discussed was the sensitivity to changes of the fine structure constants, which would be the Coulomb energy. Now, if you refer to the mass of the proton, I need to admit I have not really thought about it in detail. I expect a little bit it would be even more difficult to relate the change of the energy to the mass of the proton in terms of the coupled nuclear system, and in fact. The proton mass also enters basically into the hyperfine structure of the cesium clock. So there is some relation already. So I cannot really give you a precise answer. I think what will be the experiment should be done to really compare different clocks based on hyperfine transitions, on electronic cross structure, and on maybe on molecular structure and on this nuclear. And then from doing several of these comparison experiments, one could try to disentangle what kind of changes or interactions may occur.
Sorry, I may have missed this. My question concerns the inhomogeneous broadening of the transition, let's say in thorium and calcium fluoride. When you dope it, do you see inhomogeneous broadening, and how do you control it?
Yeah, that's a very important and I think still partially open question. So the GILA team has made important experiments quite recently and just published a paper, I think, on it. So what is expected is there's one mechanism is a spin-spin interaction of the thorium with the fluorine in the crystal. And it may lead to what theory predicts was like it would lead to like 100 hertz or so broadening. Maybe it's bigger. There's also an effect of changing the electron density interacting with the nucleus. So the GILA group has studied the temperature dependence of these lines. I think these are still open questions and certainly very interesting to investigate in order to understand more about the interaction of the nucleus with the crystal lattice. Also, in addition, there would of course also been a relativistic Doppler effect expected in the present experiments in the range of 100 hertz. So for the highly accurate solid state nuclear clock, I would expect one needs to go to liquid helium to maybe get to a zero phonon regime and to freeze out these interactions as much as possible.
Thanks.
Do you think you can build this clock on a chip?
The physical system itself, so the small solid, of course, yeah, I think that's very close to being nano integrated. The critical question then is the laser system, and the present laser system that we have been using, I've always called it tabletop, but it's a table of maybe 5 square meters or so. But going towards diode lasers and finding nonlinear optics, I think I hope it could be made small, maybe being somewhat modest, I would say, or not modest. Going to the liter scale or so would be a target that could be realistic within a few years of development, maybe. Relying very much on the laser technology, it is a critical point.
Thanks for the talk. So you mentioned the temperature. So the change in the lines related to the temperature in the nuclear clock. So in the optic clocks, from my knowledge, there's the temperature. So the lines are sensitive to the temperature too. So I'm wondering which one is more sensitive to the temperatures.
What I was referring to was the relativistic Doppler effect, time dilation, which just scales with the kinetic energy, and that is of course inside the crystal. It would be with trapped ions or so. It's the same for atomic clocks. Of course, with laser cooling, one can routinely reach the lower temperatures, millikelvin or so, not so easily available maybe as in the solid. So that would be the relativistic temperature dependence. And then in optical clocks, there is another effect that is that one needs to consider. It's the interaction with blackbody radiation. If in many of the optical clocks one has ultra cold atoms, but then the apparatus around it, the vacuum chamber, maybe is at room temperature, and then one needs to take the interaction of this blackbody radiation with the atoms into account. And this effect would effectively be absent for the nuclear clock because the infrared blackbody radiation would have no dipole moment to couple to in the nucleus. So that would be one of the advantages, where the nuclear clock would offer the higher accuracy. Not for the relativistic effects. The relativistic effects would be, of course, the same for the electronic or the nuclear transitions.
Thank you.
Let's thank Ekkehard again for a great talk.
Thank you.
Yes, so our next speaker is Jie Shan from Cornell University. There's been a lot of really amazing work lately in moiré superlattices and in just general superlattices from lattices, and we're happy to hear about her work today.
Thank you. So it is really my honor to be part of this session. thank you, Ken, for inviting me to this special Kavli Symposium. So today I'm going to talk about moiré materials and use moiré materials as a new platform for correlation and topology. So what are moiré materials? It has been known for centuries that they're layered materials which are bound together by a very weak van der Waals force. As such, those materials can be isolated into a stable unit of single atomic thickness. And for the same reason, those materials can be stacked back with arbitrary composition nearly into heterostructures, like what I'm showing here. And we have very clean surface, very stable structure. Furthermore, now, if you put the two layers of materials with small difference either in orientation or lattice constants, you are going to get this interference pattern. So what we are looking at now is the scanning transmission electron microscope image of two monolayers putting together. One is moly telluride, another is tungsten selenide. And each layer has this honeycomb lattice. So basically, you have two honeycomb lattices with slightly different lattice constants. So you see individual atoms, and on top of that, you also see this interference pattern with the triangular lattice. So this is the reason why we call this moiré. So this is the interference of two atomic lattices. So the period is highly tunable. So what this graph shows that you can change the twist angle and change the period of this moiré by orders of magnitude. So this is for two identical monolayers, and this is for two different monolayers, at a very small angle. The constant now is pretty much determined by the lattice constant difference. So now we have this new crystal. So if the parent material is either semi-metal or semiconductor, so you can think of electrons just moving in this new periodic quantum potential. So now let's take a look at some consequences. I want to mention that this new type of crystal gives us this new length scale between a real atomic crystal and, for example, also synthetic crystal made by cold atoms in optical lattices. So this new type of crystal shares the same advantage as this cold atoms with a lot of tunability. At the same time, it shares advantage of this atomic crystals that you can cool it down to temperature orders of magnitude lower than the characteristic temperature of this material. So you can study the ground states in this type of crystal very comfortably. So this is the beginning of the story. So back in 2018, Pablo Herrera's group at MIT demonstrated that once you put the two layers of graphene at an angle very close to 1 degree, and then if you sprinkle this crystal a little bit with charge, so for example, if you put something like 1 electron per 100 nanometers square, then you get an insulator. And you put a little bit less or put a little bit more, you get a superconductor out of a very innocent material made of carbon, the graphene. So this really opened a new way to study condensed matter physics. And since this first work, people have studied marine materials made of graphene, semiconductors, and many, many other structures. And we have been able to demonstrate pretty much every single many-body or topological phases found in condensed matter physics in this moiré structure. Furthermore, there have been new phenomena, new states. For example, the fractional quantum Hall effect at zero field, or fractional quantum spin Hall effect in the marine materials that have not been observed in any real atomic crystals. So what I would like to do in my remaining time is just to give you a kind of overview of the general features of this class material emerging from this moiré length scale. Then I'm going to introduce the specific class of marine materials built from transition metal that are carbonized semiconductors. Then I'll give you a couple of examples of what this material can do. So the first thing I've already mentioned, if the moiré period is much larger than the atomic lattice period, then you can separate the length scale, and accordingly, you can separate the energy scale. So for low energy, you can just think about this as block electrons from the parent material, semi-metals or semiconductors, moving in this periodic moiré potential. So now you have this new periodicity. The way we like to do in condensed matter physics is to think about this in terms of energy bands, and that's energy as a function of momentum. So with this new periodicity, what we effectively do is just to fold the zones, fold the bands. Now this original Brillouin zone becomes moiré mini Brillouin zone. So you fold this band many times. So this is exaggerated. So this is a few percent of the size of the original size of the momentum space. So large periods immediately implies that we have very strong electron-electron interaction, and it's a good place to look for strongly correlated effects. And to see this, I just have this hand-waving argument here. So the kinetic energy scales as momentum square or distance inverse square. A Coulomb interaction scales as 1 over distance. So the ratio of the interaction to kinetic energy scales linearly with distance. So for real materials with distance on the order of the lattice constant, nanometer or sub-nanometer, this ratio is on the order of 0.1 typically. So it's not strongly correlated, but for marine materials, the period is on the order of 10 nanometer. Immediately, you can get this ratio to be above 1, and you get a strong electron-electron interaction. So you can also think about this in terms of bands. So strong electron interaction, that means kinetic energy is quenched. So we have very flat bands. So every time when we have flat bands, we have a strong electron-electron interaction. The system is highly tunable. So you can change the composition of the layers. You can change the twist angle, which will change the lattice constant. And that will change the kinetic energy because that determines the hopping between the lattice sites, which decreases exponentially with the lattice constant. And you can also put this into a field effect device structure. So now they are just look like planar capacitors. So if you apply voltage on one of the gates, you can apply vertical electrical field and at the same time push charge into the moiré system. So now with two gates, top and the bottom, the two knobs can change the vertical displacement field and the doping density in the material continuously and independently. So here's just an example. Because the density of the moiré atoms is only about 1 part of 1,000 of a real atomic density, we can actually use the capacitor structure to put quite a lot of electrons to fill up the first few bands. So every time when you completely fill one band, equivalently you are changing the chemical element, from helium, hydrogen to helium, to other elements. So this is an easy way to tune the different characters of those atoms. And the system is simple. So here's just an example when we study, for example, the Kondo lattice problem, which is the interaction between a lattice of local moments, local magnetic moments, and free electrons, let's say. Conventionally, you would use, some material, like what I'm showing here, with a very complicated band structure. You see spaghetti of bands, and hopefully, near the Fermi level, you will be able to find some flat bands, and that will give you localized moments, and also dispersive bands, and that will give us itinerant electrons. So now with the marine materials, we can find something called a insulator, which I'm going to talk a little bit later. So basically, you put one particle per unit cell, and because of a strong Coulomb interaction, they like to localize. And in a band picture, they just form very flat bands, and those are due to interaction. So those are localized magnetic moments. And then you can bring in some dispersive bands and make them interact with each other. And this is quite controllable, and it's much, much simpler than what we can get from this bulk crystal. So in the remainder of my talk, I'm going to focus on this class of material, and this is a semiconductor. It's made of transition metal sitting in the middle and the charging atoms on top and the bottom. And if you look from top, it forms this honeycomb lattice, but this side and that side are occupied by different atoms. So you have asymmetry here. So if you look at the momentum space, you also get a honeycomb, but rotated. And the band's edges, the minimum conduction band and the maximum of the valence band occur at the corners. We call it K or K prime or minus K, those corners. And because we have a transition metal, it has a very strong spin-orbital interaction. So the bands are spin split, especially if you look at the valence band. So this splitting can be as large as hundreds of millielectron volts. So if we look at the small energy scale, you can forget about the high energy bands. So you just have, those two at the K point and the K prime point at those two corners of the Brilliant zone. And the system has time reversal symmetry. So that means if I change momentum from one corner to the other corner, in order to preserve time reversal symmetry, spin has to flip. So spin and the valley here are locked together. So if I just look at these bands, I have double degeneracy, and that's the valley and the spin together. Okay, so now let's just put the two of those crystals together. So for example, in this pair, if I don't twist, then I'll get a triangular lattice, with lattice constant on the order of 10 nanometer. And they align energetically like this. So if I look at where holes are going to go, they mainly will be going to the tungsten layer. And if you look at this, the other bands are very far away. So if we look at the small doping, we'll be studying mostly this band, and it's flat, right? So the problems of electrons or holes, in this band, how they interact and how they behave, can be projected to a known problem, and this is the Hubble problem. So we have a Hubble problem for triangular lattice. So at the simplest level, we can have just the two terms in the Hamiltonian. So first term describes the hopping from one site to another site with this T parameter. And this tends to delocalize the charge. And the second term is Coulomb interaction. We just consider the simplest case, the energy price one has to pay in order to put two charges at the same site. And this U tends to localize the charge because, they don't want to move. Once move, you may have two particles end up at the same site, right? So this Hubble model has been around for quite some time and has been very useful. This is considered as a model to describe high temperature superconductivity, magnetism, also metal to insulator transition. So in this particular case, T is on the order of 1 to 10 millieV, and U is on the order of 100 millieV, and it's determined by the size of the Werner orbital, which is about 1 nanometer. And those parameters are highly tunable. So you can change, for example, the lattice constant, which will tune T. You can change the dielectric screening, and that can change U. So we can actually tune through the ratio of those two parameters. So here's one of the first studies. Look at this moiré structure while tuning the doping density. So at the filling equal to 1, that means 1 particle per moiré unit cell. And because we have the double degeneracy, so this means the band is half filled. So without interaction for half filled band, we expect to see metal. But in this case, we see very good insulator. And this is at a filling equal to 2 when the first band is fully filled, and this is expected, a band insulator. When you lower the temperature, resistance shoots up. So this background is due to a contact issue. So what's happening is that when U over T is very big, the Coulomb interaction actually breaks this into two bands, the lower Hubble band and the upper Hubble band. So energetically, this is more favorable. So now we have one band completely filled. Fermi level is in the gap. So this is an insulator, but it's not a usual band insulator. This is this is a correlated insulator, and it's a Mott insulator. But to really show it's a Mott insulator, we have also studied the magnetic properties. So what this curve shows is the magnetic susceptibility as a function of temperature. And then you can see that the magnetic susceptibility is very well described by something called Curie-Weiss law. So it's just showing here. So this is a typical behavior for localized magnetic moments. And the sign of the Curie-Weiss temperature theta is negative. And negative sign means there is anti-ferromagnetic interaction between the localized moments. So this is due to super exchange interaction between the spins. So one possible ground state for the system is a 120-degree Near order antiferromagnet. So as I said, you can tune the ratio of U and T. So in this case, we switch now to a smaller lattice site. Now T is bigger because hopping is more important now. And you can also tune this displacement field, which tunes the ratio of T over U. So for small displacement field, we see that resistance increases as temperature decreases. So this is insulating. Now, if you're to tune to a slightly different displacement field, instead of insulating behavior, now we becomes metallic. So this is insulated to metal transition, and it covers many orders of magnitude in terms of resistance change. And this transition is continuous. And this is one of the first examples that allow us to study the metal to insulator transition induced by interaction. Okay, so now the question is, what happens when we have partially filled first band, but the filling factor is now 1, maybe smaller than 1 or maybe bigger than 1. And we saw there's some difficulty in doing the transport measurement due to the contacts. So we developed this sensing technique to look at the insulating states without measuring the resistance. So here's the idea. So imagine we have a piece of a semiconductor, but very thin, 1 atomic layer. And if you excite it optically, you're going to generate electron hole, which are bound, we call the exciton, right? So this exciton, you can think of it just like a 2-dimensional hydrogen atom, and it has this Rieberg states, the fundamental exciton, 2s, 3s, so on and so forth. And this is something that we have measured experimentally. And that the exciton binding energy is also very big. And the reason why it's big is because, we have a charge with not much screening. So dielectric screening is much reduced in 2D. So the exciton binding energy is on the order of hundreds of millieV. And that also means they can interact with photon very, very strongly. So now if I place a 2D electron gas system or some sample I want to measure just in near vicinity. So if this system is insulating, then it wouldn't introduce a lot of dielectric screening to the excitons I want to study, right? So in that case, if I don't see a lot of screening, that means, this is a good insulator. However, if this material sample now is metallic, that means it will introduce a lot of screening. Then I'm going to quench the intensity of my exciton and also the binding energy. So because the exciton bore radius changes, with the S number, the N number here, so I'm going to focus on this 2s exciton because it's a little bit bigger, so it has a large sensing range, and also it's strong enough to be detected optically. So let me just show you some results. So this is the response of exciton sensor very close to this moiré header bilayer. And what I'm doing is tuning the gate voltage, which tunes the doping density. And this doping density can also be converted to the filling factor. So for example, here corresponds to charge neutral moiré. And here corresponds to 1 electron per moiré, and here is 1 hole per moiré. So the color here shows the spectrum of this 2s exciton. Whenever you see blue, that means we have a strong 2s exciton response, and this also shows the energy. So when the material is charge neutral, I have very strong exciton response, and this is expected because dielectric screening is the smallest. Now get to filling equal to 1. This is our MAR insulator. So also not a lot of screening. And in between, the genetic filling, this exciton is significantly quenched. And that means we have a broad filling range where you have metallic states. But what's interesting is that on top of those metallic states, at those fractional filling, those are commensurate filling factors, you see insulating states pop up. So what are those states? So they turn out to be some kind of electron crystals. And the reason is because, not just the on-site Coulomb interaction is important. We also have a long-range Coulomb interaction in real material system. So they tend to push electrons away from each other as far as possible, so they can stabilize the electrons and form different crystals. And those crystals have their own character, magnetic properties and other properties. But, so I'm not going to get into the details of this. Okay, so let me just draw a phase diagram of this Fermi-Hubble plus long-range Coulomb interaction. So this is the knowledge from 2022 when we wrote the review article. So when you change the filling factor along the x-axis and also change the bandwidth along the y-axis, you can scan through MAR insulator, different kinds of electron crystals. Some of them breaks rotational symmetry, So a burning question is, where is superconductivity? So I mentioned that this is a model people like to use to understand high temperature superconductivity. And for a long time, people have been searching for superconductivity in this particular system. And finally, earlier this year, 2 papers, one from our group, one from Columbia group, showed that indeed there is superconductivity. So for example, in the Columbia group, superconductivity emerges near some kind of antiferromagnetic metal, but there's no insulating state. And in our study, we studied a smaller twist angle where correlation is stronger. We still have insulator, but at the tip, when insulator is killed, we have superconductivity. So whether we can put superconductivity on this figure it still needs some work to do. What is the nature of this insulator, whether this is real MAR insulator or some other insulator. So this needs to be done. So next I would like to switch gear and tell you another example where correlation and topology can work together to give rise to new states. So we look at this simple example, right? When we have a single isolated band, then, it's just the correlation physics. There's no topology, so this is a topologically trivial case. However, if you can somehow couple 2 bands, turn on some kind of a complex hopping, you can, in principle, turn those bands from topologically trivial to topologically non-trivial with non-zero Chern number. And this Chern number is topological invariant, describing the winding of the eigenstates in the momentum space. So let me just give you kind of a pictorial view of how this non-zero Chern number can emerge. So this is a work by Alan McDonald's group back in 2019. So in this theoretical work, they consider small twist angle moiré telluride bilayers. So you have one honeycomb from one layer and another honeycomb from the other layer. So this is in the momentum space. So this is the minium Brillouin zone. So now there are different stacking order, right, in this twisted bilayers, as we saw in the TEM image. So you can have metal sitting on top of metal, metal sitting on top of caucasian atoms, caucasian sitting on metal. So even the potential difference between the two layers for the whole on average is 0, but on the moiré scale, you still have spatial variation. So somewhere you have higher potential for one layer, somewhere you have higher potential for the other layer. And that's what this color shows. So the arrows actually shows the interlayer moiré potential. And you can see those kind of the direction, like the flow of these arrows, they just look like a skyrmion lattice, like what we see in magnetic materials. So electrons in the system actually sees this moiré potential looks like a skyrmion lattice. So they are effectively the magnetic fields, experienced by the electrons. So for example, this point is like North Pole, and the other point, it will be the South Pole. So then if electrons are located here, you get the non-zero Chern number. So become topological. So in this particular case, for the K-valley states that has spin up, the Chern number is -1. And for the minus K spin down, which is the time reversal copy of it, will have an opposite Chern number. So because of the skyrmion lattice structure, now we have topological bands. So what's going to happen if you have topological bands? So now imagine that we have a filling factor 1. So I have those two bands, one from the K valley with spin up. The other one is from the minus K valley with spin down. And both are half fill, right? Because those bands are flat, interaction is important. So energetically, it's more favorable to break the symmetry. So we have one band fully filled and the other band empty. And now the Fermi level is in the gap. So this is an insulator, but it has this very peculiar topological property. So it's a bulk insulating, but you have edge states. And this edge state, is pretty much the same what people observe in quantum Hall when you put 2D electron gas under a very large magnetic field, right? And this is also called a Chern insulator or zero field Chern insulator. So what happens at a filling equal to 2, you just have your chemical potential right here. So in that case, you have two copies, spin up electron running one way, spin down electron running the other way. So this is something called a quantum spin Hall effect. So the bulk is insulating and you have the this helical edge states. So both quantum Hall effect at the zero magnetic field, which we would call a Chern insulator, and the quantum spin Hall effects have been observed in all kinds of different moiré materials, including graphene, TM transition metal dichalcogenide, and also rhombohedrographene on HBN. So what's so remarkable is the recent progress studying twisted moiré telluride and also graphene pentalayer on HBN. in addition to this quantum Hall at the zero field at the integer filling, we've also observed quantum Hall at the fractional filling. So this is a work from Xiaodong Xu's group at Washington University for twisted molybdenite, and this is a work from Long Ju's group from MIT on pentalayer rhombohedragraphing on HBN. So let me just show you.
Jeez, you're out of time.
Finish, right? Okay. Okay, so let me just quickly walk through this and then I'll finish. Okay. So this is a thermodynamic study of the FCI from our group. So what this map shows is incompressibility of twisted molybdenite. Every time when you see something red, that means, you know, this system is insulating. So we see insulating system at filling 1, 2/3, 1/3, 1/4. And you can determine the gap size and so on and so forth. And it turns out those states are also magnetic, ferromagnetic. They break time reversal symmetry. If you apply magnetic field, they disperse with magnetic field. And this follows the strata formula. When you apply magnetic field, the flux can pump the states. So now it disperses with magnetic field. You can determine the slope, and those two quantum numbers tells you how to classify the states. So indeed, the one at 2/3 disperses with slope 2/3. So this is a fractional turn insulating state. And this is equivalent to measuring directly the edge states through transport. So you look at the Hall conductivity or Hall resistance, it reaches to quantize the value of, you know, h/e² divided by this value. Okay, so I'm running out of my time, so let me just skip this. I just quickly show the summary. So I hope I convinced you that the semiconductor moiré materials are a simple and a tunable platform to explore correlation and topological phenomena. And I give you a couple of examples I'm not going to read. And then very importantly, I would like to thank my group at Cornell University, my collaborator, King-Fan Mak, who co-supervised the group, my theory collaborators, people provide the crystals and funding sources. Thank you for your attention.
I believe we're out of time for questions, so we'll move on. Thank you, Jie. Thanks to you again.
Sorry.
Our last speaker of this session is Nicole Yunger Halpern, who's, you know, I guess my favorite quantum steampunk, but also when I was thinking about something to bring together March and April, this idea of quantum simulations of quantum thermodynamics on lattice gauge theories sounded perfect to me. So, Nicole.
Great. Thanks for the introduction. Thanks very much for the invitation to speak here. I'm very honored. And I would appreciate it if everyone would do me a favor and stand up. Place your fingers together, put them in front of you, and stretch. This is a long session. We've been setting a while. Okay, good job. You may sit down. Great. Now that we're all awake, we might notice that quantum thermodynamics appears in the title of this talk. Quantum thermodynamics is a subfield that has, or a field that has bloomed over the past 15 years, but not everybody is familiar with it, so let me give a little introduction. I need to start with thermodynamics. I find that although we all study thermodynamics as physicists, a lot of us don't remember what it actually is. So it's the study of energy, forms of energy such as heat and work, and transformations amongst those forms. Thermodynamics was inspired by the Industrial Revolution. Steam engines were transforming civilization. People wanted to understand how efficiently they operated. These practical questions led to fundamental questions, for instance, about the arrow of time. What is quantum thermodynamics? Well, we know from, say, quantum computing and quantum cryptography that quantum phenomena such as entanglement can enhance information processing tasks. Just as there are information processing tasks, there are thermodynamic tasks, such as charging batteries and cooling systems. So we can explore how quantum phenomena can enhance thermodynamic tasks. Second, the laws of thermodynamics were developed by people who had in mind steam engines, very large classical systems. So we can use tools from quantum information science to see how these laws extend. For instance, we can use quantum information theory to prove more precise, more detailed variations on the second law of thermodynamics. Finally, we can look at a system that's processing energy and ask, what behaviors can this system exhibit only if it's quantum? So we can use thermodynamics to understand better the division between the classical realm and the quantum realm. I see these three questions as guiding questions of quantum thermodynamics. I had a conversation with people in a quite different fields, high energy and nuclear physics at the University of Maryland, and they told me that some of their favorite systems, lattice gauge theories, have big thermodynamic problems. I should give a little bit of a review of gauge theories. These are prevalent across high energy physics and nuclear physics, as well as condensed matter. A favorite simple example is electrodynamics. A gauge theory has a local symmetry. For instance, electrodynamics has a U1 symmetry. And a gauge theory obeys Gauss's law. Gauss's law is a local constraint. It interrelates matter at some site with the gauge field around the site. In an electrodynamic example, suppose that we have a charge at some location. There's an electric field around the charge. And suppose that we take a surface that encloses the charge. We perform a surface integral. The result is related to the amount of charge inside. So again, matter at a site is related to the gauge field around the site. Now, what about lattice gauge theories and quantum lattice gauge theories? Well, lattice gauge theory has a discretized spatial degree of freedom. The matter lives at the sites, which I will index with little n. The gauge field lives on the links between the sites. How does Gauss's law manifest in a quantum setting? Well, the whole system is governed by some Hamiltonian. There are Gauss law operators, Gn, associated with the sites. These operators are conserved by the Hamiltonian. These operators define the physical states that are accessible to the actual physical system. The physical states are eigenstates of all of the Gauss law operators, associated with some eigenvalue little g. For instance, if we're talking about a gauge theory in the standard model of particle physics, then nature gives us the value of little g. These physical states are highly entangled. So relatedly, every degree of freedom, or the degree of freedom at each site, because of Gauss's law, is closely related to the gauge field around that site. But in turn, again, by Gauss's law, that gauge field is closely related to the sites on the other side, and those sites are closely related to the gauge degrees of freedom on the other side, and on and on. So we get this huge, interrelated, entangled mess of degrees of freedom. It's not easy to say that we have some independent degrees of freedom over here that we can call the system of interest, and some independent degrees of freedom over here that we can call the environment. And so it's not so easy that we can, or it's not so easy to say that there is heat exchanged by the system over here and the environment over there. And more broadly, how can we define a quantum thermodynamics for this kind of a mess of a lattice gauge theory? People in this session might care about lattice gauge theories in part because they are quantum simulable. They describe fascinating things like the early universe and the results of heavy ion collisions. Those systems can be difficult to model on classical computers, so people are looking to quantum computers to simulate them. Now, thermodynamics is kind of a queen of the physical theories. We expect all the other physical theories to obey thermodynamics. So if in lattice gauge theories, some theory, we can't even define very basic concepts of thermodynamics, such as work and heat, then we are in trouble. So I had the privilege and pleasure of working with a wonderful team of people on precisely this problem. Here's where I'd like to go in the rest of this talk. First, I'll introduce the toolkit that we used from quantum thermodynamics to address this problem with lattice gauge theories. I'll specify a protocol we'll focus on. It's a protocol that can be performed in quantum simulators with systems that are simulating lattice gauge theories. We'll use that toolkit in order to define the work and heat exchanged within a lattice gauge theory system during that protocol. And then I'll show an example application of our framework to a Z2 lattice gauge theory. And we'll see that the work and heat definitions that we came up with are useful because they point out a phase transition, so some large-scale phenomenon. And I'll also point out there are many opportunities for future work. Our toolkit is strong coupling quantum thermodynamics. If anyone wants an explanation of the aesthetic of this talk, feel free to answer during the question and answer session. Strong coupling quantum thermodynamics is based on a lot of earlier work. We can understand it by going to the opposite extreme and thinking about weak coupling thermodynamics, which is much more commonly invoked. Here we think about a system and a reservoir that exchange heat, and also perhaps some external control system performs work. The whole system is governed by a Hamiltonian that breaks up into a system term, reservoir term, and coupling. It makes sense to define as the average total internal energy, the expectation value of this Hamiltonian. And we assume, in the weak coupling context, that this interaction term contributes negligibly to that total internal energy. One reason, often, for making this assumption is that the size of the coupling, according to some appropriate matrix norm, will scale only like the boundary of the system of interest because most interactions are local and so occur just across the boundary between the system and environment. In contrast, the energy of the system of interest grows like the volume of the system of interest, and the reservoir's energy grows or is even larger, and so the coupling will be negligible in comparison. In this case, it makes intuitive sense to define the average internal energy of the system of interest as the expectation value of just the system Hamiltonian. If the reservoir is at some inverse temperature beta, then very often we will expect the system state to end up in a thermal state at that inverse temperature. And I'll use pi to denote equilibrium states. However, our system of interest might be small, for instance, if it's quantum. So this justification for weak coupling can break down. In this case, it isn't clear how we should break up the coupling energy between the system and the reservoir, or to what extent we can break it up. So we need strong coupling quantum thermodynamics. The strategy here is to define energetic quantities, often by analogy with a weak coupling case, and then justify these definitions by showing that they obey laws of thermodynamics, so they are reasonable definitions to use. Again, we have a system and a reservoir. Let's suppose that the whole thing is in a global Gibbs state, which I'll denote by pi S union R. There are a couple of justifications for this assumption. Feel free to ask during the question and answer session. To get the reduced state of the system of interest, we trace out the reservoir, we get pi S, and we are free to write that this state equals some function. It looks like a thermal state, but it has this funny HS star playing the role of a Hamiltonian. This HS star is called the Hamiltonian of mean force. This formula for it looks a little complicated, but rest assured, in the weak coupling regime, this formula reduces just to the system of interest Hamiltonian, and I can show you a proof during the question and answer session if you want. Now, this Hamiltonian of mean force from quantum thermodynamics, we found, is very closely related to an important object in many-body physics. That important object is called the entanglement Hamiltonian. And for our story, it's important because in many cases, one can experimentally measure it efficiently. We're going to take advantage of that notion later. And this ZS star, I'm going to define as normalizing the state. It makes sense intuitively to define the average internal energy of the system as the expectation value now of this Hamiltonian of mean force. This definition naturally incorporates some contribution from the coupling. We can also, if we like, define a free energy function. And suppose that our system undergoes some process. Let's denote by W the work absorbed by the system during that process, and denote by Q the heat absorbed. And when I talk about energy quantities during this talk, I'll implicitly be referring to averages. Whenever we talk about these thermodynamic quantities, we need to talk about the laws of thermodynamics. Probably all of us recognize the first law of thermodynamics. The change in a system's internal energy can be parceled into work and heat. The second law tells us about how much work we have to spend if we will change the Hamiltonian of the system of interest. If we, at least classically, in ordinary thermodynamics, if we change the Hamiltonian extremely slowly so that the system is always in equilibrium, then the amount that we have to, of work that we have to spend is minimal. It's the difference between the free energy associated with the final Hamiltonian and the free energy associated with the initial Hamiltonian. In general, the work we pay is at least this free energy change. Great, I've stated these laws of thermodynamics. I've introduced notation for work and heat, but I haven't actually given you definitions for work and heat. How we define work and heat depends on what protocol we're performing, during which the system absorbs work and heat. So we need to pick a protocol. Our guiding principle will be, we don't want to upset our experimental friends, so quantities of interest should be measurable via access just to the system of interest. We don't want to have to measure the giant environment. We're going to focus on a sudden quench process. The Hamiltonian will change instantaneously, or at least extremely quickly. Such processes are implemented on many quantum simulators. Again, we have a system, we have a reservoir. Now there is a total Hamiltonian that can change in time because the system component can change in time. This process happens over an extended period of time with 3 steps. The first step is the preparation procedure. Suppose that the Hamiltonian, the total Hamiltonian, is in some initial state associated with T0 minus because the system of interest Hamiltonian is in this initial form. Again, we suppose that the whole system begins in a Gibbs state, which has the exponential form we expect. And we get the system of interest state by tracing out the environment. We get rho initial system. The second step is the quench. So the system of interest Hamiltonian changes from its 0 minus form to its 0 plus form. And accordingly, the total Hamiltonian changes from its 0 minus to its 0 plus form. This quench happens so quickly, the state doesn't have time to change. It does have time to change during the third step, equilibration. We suppose that the system and environment just sit there for a long time so that they end up, again, in an equilibrium state. This equilibrium state looks like the initial states, except we have an F and a 0 plus instead of an initial and a 0 minus. We get the system of interest final state by tracing out the reservoir as usual. Now that we've specified a protocol, we can define the heat and work absorbed by the system during this protocol. Again, we want to be able to infer energetic quantities of interest by measuring just system degrees of freedom. We don't want to have to worry about the environment. We'll define the average work performed on the system as follows. In general, work is controlled energy that gets into the system because we're tuning some control parameter. That tuning happens entirely during step 2, the quench. So the work, on average, is a trace. It comes from taking the difference between the final and initial Hamiltonians, and during the quench, the system state remains constant. We can also define the dissipated work. Again, in the ordinary classical case, this difference between free energies is the minimal amount of work we have to spend. So the dissipated work is the extra work that we put into the system to roil the system up. We expect this dissipated work quantity to be experimentally measurable, feasibly, because of that connection I mentioned earlier. The Hamiltonian of mean force from quantum thermodynamics is related very closely, we found, to the entanglement Hamiltonian from many-body physics. And that latter object is often experimentally measurable, feasibly. Now, what about heat? Heat is energy that the system exchanges with the reservoir. And that heat exchange happens during the third step of the protocol. So the average heat, since it's an average, is a trace. Now, the state of the system changes during this step, while the Hamiltonian remains constant. I've posited these definitions. I need to convince you that they're reasonable definitions. We did that in a follow-up paper that appeared on the archive recently. These definitions obey the first and second laws of thermodynamics. Now, we can remove the mask from lattice gauge theories using our strong coupling quantum thermodynamics background and see that this complicated lattice gauge theory system is nothing more than a strongly coupled quantum thermodynamics system. Remember, Gauss's law ensures that the system remains in the physical subspace. We can formulate Gauss's law as a penalty term added on to the Hamiltonian. Many people have written about this. So we can take our lattice gauge theory Hamiltonian, add on this penalty term. This F is a function of the Gauss law operators. Remember, a Gauss law operator couples matter at a site to the gauge field near the site. That is, it contributes to the coupling term in our thermodynamic Hamiltonian, or thermodynamic decomposition of the Hamiltonian. And if this Gauss law term is to restrict the system to the physical subspace, the Gauss law term should be large, so it can affect strong coupling. Here's the takeaway so far. We can see a lattice gauge theory system in one of two ways. If we're not thinking of the Gauss law term as a penalty term in the Hamiltonian, then the lattice gauge theory system looks like this hopelessly interconnected mess of many degrees of freedom. But if we think of Gauss's law as a penalty term in the Hamiltonian, then we can see a lattice gauge theory as having a system and an environment that are just strongly coupled in the sense of strong coupling quantum thermodynamics. As a result, we can use our definitions of heat and work from strong coupling quantum thermodynamics. So we can identify the heat and work exchanged within a lattice gauge theory. How do we do that? Here's an example, and I'll also show why doing this is useful. Our example is a Z2 lattice gauge theory. We have a 1 + 1 dimensional lattice. The matter consists of hardcore bosons. They live on the sites that I index with little n. The total number of sites is big N. Associated with each matter site is a set of Pauli operators sigma AN. The gauge field also consists of hardcore bosons. These live on the gauge links. They're associated with Pauli operators tilde sigma. Since we're theorists, our system has periodic boundary conditions, and this model was adapted from a model that involved fermions, and there are some artifacts of that earlier model in the terminology I'm going to be using. Here's our Hamiltonian. It has an electric field strength term for the gauge field. So sigma tilde x is our electric field operator. Epsilon is the electric field strength. There's a hopping term. So matter can hop from site n plus 1 to site n. As it does so, it hops through the gauge degree of freedom in between and flips the electric field. J is the hopping frequency. There is a mass term for the matter. This -1 here is staggering. It is an artifact of that earlier fermion model. There's a chemical potential for the matter. And there's this constant that is in the model for just numerical stability. It has no physical implications, so don't worry about it. The Gauss law operators for this model look like this. We can break down this operator. This exponential factor acts on the matter on the sites. The sigma plus sigma minus counts how many particles are at site n. And the sigma tilde x's are the electric fields on either side of the site. We can understand this operator further by supposing that there is no matter. This Gauss law operator is essentially just the x operator on the gauge degrees of freedom, which generates the Z2 symmetry group, and that's why we're calling this model a Z2 lattice gauge theory. We need to incorporate our Gauss law operators into the Hamiltonian in a penalty term. So G sub n is the Gauss law operator, and kappa is the strength of this term. You've already seen all the other terms in the Hamiltonian. Now we need to partition the lattice into the thermodynamic system of interest and the environment, and we similarly need to partition the Hamiltonian into a system term, a reservoir term, and a coupling. Honestly, the process is kind of tedious, so I'm not going to show you it, but again, feel free to ask about it during the question and answer session. What happens during our quench process? We take this chemical potential, it's initially 0 at all sites, and then within the system of interest, we raise its value. This model we have evidence for 2 phases of the model. I'm not going to show you the order parameter. Again, feel free to ask during the question and answer session. Suffice it to say, the phase of this model is controlled by the chemical potential. So if the chemical potential within the system of interest is large, the system is in one phase. Terminology is an artifact from the earlier fermion model. And if the chemical potential is small, then the system is in a different phase. In case you want to know the values of the parameters that we used, here they are. They're order 1 to order 10. And in this case, the phase transition occurs when the chemical potential is 1.82. I'm going to show it on a plot. That's why I'm presenting the actual number. Here's the plot. Here are the results. Along the x-axis, I'm going to plot the final chemical potential, the result of the quench. Along the y-axis, I'll show the work that was dissipated during the quench, defined with strong coupling quantum thermodynamics. Here are the results. The dashed line shows where the phase transition occurs according to the ordinary order parameter. This dissipated work changes pretty noticeably at the phase transition. We can generate a similar plot for the heat absorbed by the system during the protocol. In conclusion, work and heat that we define using this toolkit of strong coupling quantum thermodynamics, a totally different field, signals a phase transition within a lattice gauge theory. So applying quantum thermodynamics to lattice gauge theories in a quantum simulable protocol seems useful for learning some large-scale property of the system. There is much to be done. What we're working on now is checking that indeed we can measure the Hamiltonian of mean force efficiently in an experiment using this connection with many-body physics that I mentioned. early results seem promising. And then we would like to convince an experimentalist, I'm looking at you, Ken, to perform our protocol and experimentally measure the work dissipated in a quantum simulator that is simulating a lattice gauge theory. We should generalize these definitions of work and heat to other processes that a lattice gauge theory system can undergo, for instance, particle collisions. We'd like to see that these work and heat definitions are otherwise useful in the context of lattice gauge theories. Maybe they signal other types of phase transitions, such as topological ones, or maybe these definitions can signal completely other macroscopic phenomena. More generally, we'd like to build a quantum thermodynamics of lattice gauge theories. In summary, we used the toolkit of strong coupling quantum thermodynamics. A key result was a relationship between the Hamiltonian of mean force from quantum thermodynamics and the entanglement Hamiltonian from many-body physics. This entanglement Hamiltonian can be measured experimentally in a reasonable protocol. We can take advantage of that result in quantum thermodynamics. We focused on a protocol that can be implemented on a quantum simulator used to learn about a lattice gauge theory system. Using that toolkit, we defined the work and heat exchanged within a lattice gauge theory system undergoing that protocol. And we saw in an example of a Z2 lattice gauge theory that these work and heat definitions can be useful. They signal a phase transition. We also saw there is much less left to be done. Thanks for your time.
All right, questions for Nicole? I loved your talk, just fascinating. I don't have any questions about the steampunk vibe, which is great, but I would love to see the derivation or whatever appendix you have on this Hamiltonian of mean force.
Certainly, thank you. It's going in the opposite direction. Sorry, I have a lot of bonus slides. All right. We said that there's a relationship between the Hamiltonian of mean force from quantum thermodynamics and the entanglement Hamiltonian from many-body physics. Here are some references about, early references about the entanglement Hamiltonian. The entanglement Hamiltonian is defined for just an arbitrary state, rho s. You take the log of the states and introduce a negative sign. A motivation for this example is the thermal state. The entanglement Hamiltonian is more or less, to within a normalization factor, the Hamiltonian of the system of interest. And again, in some cases, this entanglement Hamiltonian can be measured efficiently. So suppose that our rho s, our system of interest state, looks like a thermal state with respect to this Hamiltonian of mean force. Then the key equation is that the Hamiltonian of mean force is 1 over the inverse temperature. So the temperature times the entanglement Hamiltonian plus this free energy quantity. So the Hamiltonian of mean force and the entanglement Hamiltonian are interrelated linearly. And I have yet another bonus slide that shows how this equation implies that our dissipated work, which we actually want to measure experimentally, is it more or less consists of just entanglement Hamiltonians.
Okay, thank you.
I also loved the talk, but I'm confused about something fundamental here. In a gauge theory like electrodynamics, the Gauss law enforces gauge invariance, which is what leads to charge conservation, and it also gets rid of non-physical degrees of freedom. if you are only enforcing the Gauss law as a penalty term in the Hamiltonian, what happens to charge conservation and what happens to the spurious degrees of freedom?
We can talk about, let's say, the ideal case and the context of quantum simulators. In the ideal case, we can say that the penalty term in the Hamiltonian is so extraordinarily large that we never have to worry about the systems moving out of the physical subspace. Then we can say, suppose that the penalty term is something that is engineered to be part of the Hamiltonian in a quantum simulator experiment to try to ensure that the system, which otherwise could get out of the physical subspace, is restricted to the physical subspace. This is a subject of work that a number of other people have done. In an experiment, we can't tune that penalty term to be arbitrarily large. So there's a matter of just trying to make that term large enough. I haven't worked on those papers that propose this experimental technique and try them out, but according to my understanding, this is one of the tools that people with quantum simulators are exploring for trying to ensure that their tabletop system behaves as well as possible like a true lattice gauge theory.
Okay, thank you.
Hello. So yeah, this was a really great talk. I'm concerned you may have already answered this a few times, but I may not have grasped it. So as far as experimental techniques, in terms of experiments I'm aware of, is there any way to like either extract it directly or like use a fitting function to learn more about this from like ARPIS or pump probe spectroscopy or other common techniques like that?
I'm going to have to defer to the notion that I am a theorist.
I understand.
So let's see. The key experimental point in this talk was that we can use the connection shown on this slide between the entanglement Hamiltonian and the Hamiltonian of mean force in order to, we expect, measure the dissipated work efficiently. The entanglement Hamiltonian can be measured efficiently. It's as we see here, the entanglement Hamiltonian is essentially equivalent to the quantum state of the system. And ordinarily, we would think in order to learn the quantum state of the system, we would have to perform quantum state tomography. That takes a number of resources that scales exponentially with the system size. That is not feasible. The idea here is that in certain important cases, this Hamiltonian is well approximated by a simple parameterized ansatz. And so we take advantage of that ansatz, measuring a relatively small number of parameters experimentally in order to come up with a very good approximation to the entanglement Hamiltonian, and so the quantum state, and so ideally the dissipated work.
Thank you so much.
All right, so we're going to take one online question and then we'll have to ask questions later. I'm sorry. So the online anonymous user asks, how is the second law of thermodynamics modified by the quantum regime?
How is the second law of thermodynamics modified by?
The quantum regime.
The quantum regime.
Or is it not modified?
Or is it not modified? Okay. So there are lots of results within quantum thermodynamics that pertain to the second law. And you might find people with different opinions. In my experience, the second law is still going strong. For instance, whenever one designs or experimentally realizes a new quantum engine, something that one will commonly do is calculate or measure the efficiency of that engine and check, yes, indeed, it obeys the bound set by Carnot's theorem, which is a formulation of the second law. That's one example. On the other hand, there have been derivations of more detailed, more refined variations on the second law of thermodynamics. For example, fluctuation theorems are statistical statements The second law of thermodynamics tends to tell us about averages, but we can think about individual trials and think about the probability of having to spend this amount of work or that amount of work on a single trial. Fluctuation relations tell us about that more detailed information. There are other examples apart from fluctuation relations, but that is one set of something that perhaps one might want to call second laws.
Okay. All right, let's thank Nicole again. And then just a couple more quick thank yous. So first, let's thank all of our speakers again. And then I'd really like to thank the Kavli Foundation and the APS for giving me the opportunity to put together a broad range of science. And I hope you enjoyed this sort of, it's a small quantum world, or it's a quantum world. I was hoping to have It's a Small World song worked out by now, but I don't have it. So anyway, enjoy your evenings. Thank you.